So I will request Dr. Nikhil to kindly deliver his lecture.
Good morning, all. May we start our session? Hope with almost being at the end of session, we are almost clear of various aspects of the standardization from both angles of how we are developing the standards and how we are implementing them. From both aspects, you might have learned a lot and already with your existing knowledge and with these things. We will just take a review that exactly till now many persons might have told you Pharmacopoeia Commission mainly works with two focuses. One is acting as central drug laboratory in which capacity this training is being conducted, and another one to establish pharmacopoeia. So whatever development of quality standards, what we are doing for it, how those standards are established, everything is already discussed. So the final outcome of such development of standards, how it is being brought to public domain and how it is utilized by uh, central and state agencies, that we are going to see the shape of outcome, how it is being thought out. So that's the today's topic, introduction to our Ayush Pharmacopoeia. So though we say here Ayush, it doesn't mean all systems of Ayush because yoga and naturopathy are obviously non-drug therapy. So not having any drug, obviously there will not be any pharmacopoeia. So as of now, we have four pharmacopoeias for Ayurveda, Siddha, Unani, and homeopathy. One more system, Sovarikpa, we have recently added to our regulations. It has been recognized, which is prevalent in Indo-Tibetan border, so wherever we are having those borders. And, previously Buddhist dominated areas. So Himachal Pradesh, some parts of Uttarakhand, even in some parts of Jammu and Kashmir, Ladakh, Arunachal Pradesh, Sikkim, etc. All bordering areas to Tibet, we are having it. But as of now, there are no pharmacopoeias or any regulatory provision for Sawadistan. So we will skip for it and we will go for the rest of the things. Before entering to topic, I would just like to clear one thing for especially those who are working in the state regulatory field. One common assumption we always take that when we talk about Indian system, our whatever traditional medicine, drug means it is a plant based. It is true up to some extent because more than 80% of our drugs are of herbal origin. So many drugs are of plant origin. But what happens? We make a two-way mistake. School buses are of yellow color, but that doesn't mean that any bus on the road with yellow color will be school bus. Our drugs are plant based, but that doesn't mean that any plant in the market or available in the field will be our drug. And this is being misled in many cases. Noni and what that ginseng, many materials are coming in market. Herbal supplement, what plant-based medicine, whatever names they are giving, and they are trying it to bring under traditional medicine. Since Ayurveda, Siddha, Unani, our names, they are getting catchy attention of each person. So naturally, the market is trying to grab that opportunity and take benefit for their purposes. But we should be cautious. Have to be very strict to our definition. For Ayurveda, Siddha, Unani, we have first schedule of Drug and Cosmetics Act, where authoritative books have been mentioned. Whatever coming in those books, that only stands as drug of Ayurveda, Siddha, or Unani. It doesn't mean that any plant which is available will come under our purview. So just be cautious. And with that line of question, I will start with the actual presentation. So when we talk about quality standards, pharmacopoeias are mainly for quality standards. So whatever quality standards available in market, if we grossly segregate for drugs, we will segregate them in three parts. One will be our pharmacopoeias, another will be various databases published by government, non-government agencies, and research outcomes published in journals by various researchers. So obviously, databases and research journals are not the focus of our today's discussion. So we will focus on pharmacopoeia. There is no standard definition for pharmacopoeia. However, we are trying to understand it. The first point you can take note of each and every word in it. Pharmacopoeia is a collection of monographs where minimum standards feasible under given circumstances are drawn up, subject to revision and scrutiny. They are mandatory to ensure safety of a marketed product and to levy penalty for failure to keep beyond the limits prescribed in them. We will be describing drugs. So monograph is mono single, graph picture, entire picture of a given single drug that will constitute as a monograph. So here we are giving descriptions of one drug. Simply we can say like other books for one chapter, like here that there will be one monograph which will describe given drug, whatever under concern for that monograph. What this monograph will contain, though we call it monograph, so the word monograph as a general means that it is giving entire picture of that drug. But here we are having this as a regulatory document. So we give minimum standard. Just simply if you have ashwagandha in your hand, you can start with it to naked eye examination and you can go up to its DNA synthesis. Sky is the limit if you are talking as a pure science. But it may not be necessary and may not be possible that each and every research aspect what we are carrying out for that drug will be covered under pharmacopoeia. Pharmacopoeia strictly focuses on minimum standards feasible, जिसको कोई भी कर सके, कहीं पर भी वो एडॉप्ट हो सके, सबकी फाइनेंशियल कैपेसिटी में, सबकी सोफिस्टिकेशन में वो विद इन रीच हो, ऐसी ही चीज है. Whatever is possible for everybody, that will be taken up under pharmacopoeia. Here we have to keep that fine balance that to make it possible to everybody, we will not decrease our level of standards also. And at the same time, it should not be so much stringent that nobody can accept it. So with that balance, we are going for pharmacopoeia. That's what standards feasible under given circumstances. In given circumstances, whatever applicable and feasible, that we will take, subject to revision and scrutiny. No science will be science if it is fixed. एक बार बोल दिया और ऐसे कैसे ही जो पत्थर पे लिखा है, it goes lifelong, then it won't be a science. Then it will be just a record. So this we are preparing in scientific way for a reproducible implementation. So many times we need revision also. Many suggestions we are getting from stakeholders, and accordingly our pharmacopoeias we are making any correction. All of you are also welcome. If you find any discrepancy in any published pharmacopoeia, please let us know through official way, and we will take cognizance of the same, and that can be overcome during subsequent revision. Now, to the next sentence, mandatory to ensure safety. So, it becomes a regulation, as I told. Pharmacopoeia will be used by authorities to implement its standards all over the territory of India. So, when we talk about any regulations are grossly divided. जो भी है total हम जो इंडियन पिनल कोड बोलते थे या न्यू वन भारतीय न्याय संहिता BNS, that entire system of law, it can be distributed in three gross categories. One, what should be done, what not should be done. Second thing, if first point is violated or who is empowered to uh, see that what is to be done and what is not to be done. क्या कर सकते हैं क्या नहीं कर सकते हैं यस नो बताने वाला एक पार्ट रहेगा उसको कौन मॉनिटर करेगा कौन उसको इम्प्लीमेंट करेगा ये देखने वाला दूसरा पार्ट रहेगा. So first will be we will fix a frame. Second, we will fix a monitoring mechanism. And third one, if the second monitoring mechanism finds that the first thing frame is violated, then उस पर action क्या ली जाएगी. Just for example, अगर common example से अगर हम चले, when we are driving a vehicle on the road, we need a license. So this became first consideration that for driving a vehicle, you need a license so that it is ensured that you are sufficiently acquainted with the driving skills and you know the traffic rules also and you can implement it. So this was first portion. In second portion, there will be one road traffic controller who is empowered to verify whether you are going as per license, you are holding that license or not. If you are violating any rule, he can point it out to you and register the case. And in third case, if you are violating the rule, आप बिना लाइसेंस पकड़े गए हैं तो वो जाकर उसके लिए कोर्ट में जाएंगे कुछ पेनल्टी लगेगी तो फाइन भरना है या फिर कोर्ट केस चलेगा. So that becomes third part, means the corrective measure taken against violation. So here, if we see now pharmacopoeia in this perspective, the first two things are served in pharmacopoeia. The one thing, क्या करे क्या ना करे, what should be done, what should not be done. The regulatory standards, what should be applicable to the drug, that we will be mentioning. And the second part, the enforcement authorities, they are enabled through this document that they can verify. If a common person from outside, if asks me that what you are doing in Pharmacopoeia Commission, I simply tell them in this manner. You might be knowing the word drug inspector. They say, yes, okay, we know drug inspector. There is one drug inspector like police. Drug inspector can go and check at any place. What he will check? He will take any drug sample and will see whether it is correct or not, appropriate or non-appropriate. How he will check? That pharmacopoeia will check. What is to be tested, how it is to be tested, and which result is to be told as correct and which result is told as wrong. Like that, we will go in there. So, this was the first point with elaborate description. Second point, description of each single drug included in pharmacopoeia called monograph. Already we have discussed. So, pharmacopoeia becoming a legal binding, generally, it is all over world. If you see, it will be hand in hand, pharmacopoeias and regulatory authorities. So, whatever state or central government in every nation, whatever applicable case there over, so those governments will be having that pharmacopoeia. In many cases, government itself prepares its pharmacopoeia, but in some cases, like best example, USP, United States Pharmacopoeia. Government of United States doesn't prepare pharmacopoeia, but they have one United States Pharmacopoeial Convention. That convention prepares pharmacopoeia, and government and that convention, they have an agreement that whatever prepared by convention will be adopted by government for their enforcement. So not directly by government, but it is endorsed by government. So either pharmacopoeia will be established by government itself, or if not established, it will be endorsed by government, whatever state agency. But we don't have any copyright mechanism for this word pharmacopoeia. So many private publishers, authors, they have also used this word freely, pharmacopoeia, in their title. And we cannot have any restriction that nobody else other than us can use pharmacopoeia. So there are other pharmacopoeias also. So if you are referring a pharmacopoeia, please be cautious. Use them with whatever mentioned in it and ensure yourself that you are referring to the correct pharmacopoeia which is applicable by government as a part of law. Triple IIM Jammu, Indian Institute of Integral Medicine, they had prepared one Indian herbal pharmacopoeia with some medical association that they have prepared. Their name is Indian Herbal Pharmacopoeia, but that doesn't have any regulatory enforcement. So that cannot be adopted as a standard if you are referring for any drug. Forget about private. Government. Kerala government has published one book for Ayurveda in the name of Pharmacopoeia. Pharmacopoeia, but it is a formula. They have given ingredients and dose related details of all the Ayurvedic drugs. That pharmacopoeia book is available from the government of Kerala itself. But it is not used as a pharmacopoeia, it is not a book of standard. The word pharmacopoeia, how it is coming, is actually an ancient word of Greek origin. In Greek, the word pharmakon means drug. POI stands for make, and the last I stands for the ING suffix. So, as such word is related to drug making, and there are standard conventions that how if you are spelling a word of Greek origin to concurrent English, then what alphabet of Greek has to be made to what alphabet of English? Accordingly, that what PHARMSUPOER stands. So in India, we have different enforceable pharmacopoeias, Indian Pharmacopoeia, IP, API, Ayurvedic Pharmacopoeia of India, similarly Siddha, Unani, Homeopathy. But what happens, whatever materials we are using our drugs, let's example turmeric. Turmeric is a drug in Ayurveda, turmeric is a drug in Siddha, turmeric is a drug in Unani, it is a drug in homeopathy. It is a food material, it is a for its yellow colored pigment, it is a material of concern for the paint industry also. So for as a chemical ingredient of color pigments in it, they are used in dyes and synthetic colors. In that industry also, it is being used as an industrial commodity. It is used in food. It is an agricultural product and it is a medicine also for various systems. So what happens? Now regulatory domain changes from each. Agricultural product is governed by some different authorities in India. Food products are governed by some different authorities. If it is used as a color pigment, that synthetic industry, that will be regulated by some Bureau of Indian Standards or something. If it comes as Ayurveda Siddha medicine, then it will come under purview of Ayush. So whoever is regulating that material, they will define their standards according to their name. So you will find overlapping standards. One material available in 10 different sets of standards for their specific use. Then which one to choose based upon our intention, what we are going to use. If you want to use that turmeric as Ayurvedic medicine, the standards of Ayurvedic pharmacopoeia will apply. If you want to use it as a food material, standards of FSSI will apply. If you want to use it in a color pigment, standards from Bureau of Indian Standards, international standards, that will become applicable. Likewise, we will go. So, FSSI, ISI, AGMAR, these standards are also there in India, which are enforceable, but the enforcement will depend upon intent of the use. Next, I have already discussed Indian Herbal Pharmacopoeia. It was prepared by Indian Drug Manufacturers Association at IIM Jammu, but it doesn't have any official status. And there are many other pharmacopoeias, some private publications from Rajasthan, and as I discussed, government publications from Kerala. So, in case as far as our Ayush pharmacopoeias are concerned, Ayurveda, Siddha, Unani, and Homeopathy, 4 pharmacopoeias we have in hand as of now. And the structure is more or less in all cases. That too, when it comes to Ayurveda, Siddha, and Unani. You might be aware already that even our all regulatory process is also exactly same for all 3 systems, ASU. It is covered under Chapter 4A of Standard Cosmetics Act, and all standards are parallelly for all these 3 systems. So, the pharmacopoeia and formulary, whatever structure we are discussing, that goes exactly same for Ayurveda, Siddha, and Unani. I have taken an example of Ayurvedic pharmacopoeia because we have published more and more volumes and more varied content in Ayurveda. So are taken it as a baseline. Whatever we are discussing about Ayurveda, same will be exactly applicable in case of Siddha and Unani also. In case of homeopathy, there will be some differences, which we will discuss at the point wherever those separations are coming. So first of all, how we are developing pharmacopoeia. What is the modus operandi for it? In first stage, our pharmacopoeia committee will identify the material. From PCI mandate, we have a three-tier structure for preparing this standard. There are individual subcommittees on Ayurveda, Siddha, Unani, and homeopathy. Then at second level, it goes to main pharmacopoeia committee, and at next level, it goes to our scientific advisory board. Whatever standards we are preparing, so from these levels itself it starts, then the standard document is prepared, again vetted by all these committees, and whatever final recommendation after getting the comments from all over India also, whatever final document we are preparing, Government of India publishes them in the form of but there are two documents. One is pharmacopoeia, another is formulary. Pharmacopoeia is being used by analysts. So persons of Ayurveda Siddha background as well as persons of chemistry, botany, cognosy, microbiology. Everybody who is in the field of analysis, they will be referring to pharmacopoeia. So pharmacopoeia becomes more popular as compared to formulary because formulary is mainly referred by persons of specific system. Ayurveda Siddha Unani related fellows will mainly follow their formularies. And at some instances, maybe for 1 or 2% cases, botanists might require to refer. But the main target or audience for formulary is system related people only. So that becomes less popular. So if you hear from outside, you will hear the word pharmacopoeia much more frequently as compared to formulary. But when we are developing our standards, when we are working at government level, formulary is more important for us than compared to pharmacopoeia because formulary prepares the base and pharmacopoeia goes at the next level of the work. So firstly, drugs or formulations, whatever, we have that first schedule for Ayurveda, Siddha, Unani, and second schedule for homeopathy. Related to literature is mentioned. And from those books, we will identify some formulations, drugs, whatever, single, maybe single in some cases, maybe formulation. So whatever identified from those books, that will be taken in firstly in the book of formulary. And then after preparation of formulary, at second stage, we move on to preparation of pharmacopoeia. Here, pharmacopoeia gets divided into two parts. One series is Ayurvedic Pharmacopoeia of India Part 1 and another is Part 2. Take a simple example, Triphalachurna. Everybody knows the name. So Triphalachurna, it is mentioned in some of our classical literature. So we adopted it. So Triphalachurna becomes a medical drug of comes under our definition. It will be included in formulary. Step 1 covered. Now for step 2 of pharmacopoeia, we have two approaches. One, we need standards for the Triphalachurna as a formulation itself, and at the same time, we need standards for its ingredients. There are three ingredients in the Triphala: Harad, Bhaida, Amla. For all three single drugs, we need standards, and for formulation also we need standards. So the standards for single drugs, whatever coming as ingredients in this Triphalachurna, they will become part of Part 1 series of pharmacopoeia. And the formulation Triphalachurna itself will become part of Part 2 pharmacopoeia. The Part 1 series goes for single drugs and Part 2 series goes for formulation. So the first stage, what we are preparing as Ayurvedic formulary, with details about this formulary. It is official compendia identifying details of the formulation. Formulation, in most of the cases, we are taking formulation from our classical literature. But at some places, at some instances, where our expert committees have found that some formulations which are not there in classical literature, but they are needed to be regularized, they are also been taken into consideration. In very first pages of first part of Ayurvedic formulary of India, you can find one formulation, Ashwagandhati Layam. This was formulation, a proprietary medicine prepared by IMCOP, Cooperative Drug Manufacturing Association in Tamil Nadu. They had their one book in that IMCOP formulary. This formulation was designed and published. That has IMCOP formulary is not a classical authoritative literature. That formula book of IMCOP doesn't come under our purview. That will not come up with the definition of drug as per Act. But still, that one formulation from that IMCOP has been adopted as a classical formulation in the formula. Similarly, in recent days, you might have seen famous formulation Ayush Kadha in periods of COVID. That also came. So that was not mentioned in any of the classical books, but still we have given it place in the formulary. And since formulary is also a part of first schedule, so whatever comes in formulary, that becomes classic. It is a two-way approach. Classical drugs come in formulary, but if any non-classical thing is forcefully added in formulary, that will also automatically become classic. These two examples you can see, one Ashwagandha Layam from IMCOP and another Ayush Kadha. So what this formulary does, this formulary differs the selected formulations with descriptions of composition of drug, of whatever ingredients in it, method of preparation. That preparation method, instead of giving separately in each case, because as of now, if only we see for Ayurveda, we have published almost 1000 formulary standards. In case of Unani, it goes even up to 1300. So it may not be possible for each and every formulation to give it procedure. The formulary is a concise document. So what we do, we give a general preparation method for that given dosage form. So churna, all powders for medicine. That will be a common enlisted method will be given. And if any specific formulation is having any specific deviation from that general method, that will be described as a special note. So general dosage form related method of preparation as well as specific to the formulation they are given. Then therapeutic indications, contraindications, its dosage, and if there are any precautions, whatever mentioned in the text, they are given in this formulary. Generally, for formulary, since most of things we are taking from classical literature, to mention therapeutic indications and contraindications, we go for the indications mentioned in the book itself. We don't add anything from outside. If we have to add any new indication, you might have already handled this issue during yesterday's lecture on regulatory methods that under rules, if new indication is to be taken, whatever things that to be done. So may not be important for persons from cognoscity or chemistry, but for Ayurveda person or for Ayurveda Siddha Unani, for system related persons, these documents become very much useful. So why we need it? Because whatever literature available in classic, as of earlier days, it was okay. If I am sitting as a Vaidya, traditionally, I was preparing my medicines in my kitchen pharmacy and I was dispensing it to my patients only under my strict observation. There was no third party interference. So whatever I was preparing, I was knowing whom to give, what to give, when to give, and if it is having any problems in it, how to recover it. That everything was under regulated conditions. But now, a person harvests drugs from some place in Kerala, in Himalayas, higher altitudes, then that is marketed to some market of Mumbai. It is procured by some fellow industry person sitting in Kerala. He prepares the medicine from it. He puts it in at Amazon, and some person sitting at United States takes them, and with online consultation from a German consultant, he consumes that medicine. So it is not under one condition. So here we need that whatever we are preparing should follow some minimum standards, that the user and the prescribing physician must know what it contains. So we need these all regulations. And the available classical literature is not sufficient to give reproducibility of the content. हम घर में चाय भी बनाते हैं तो बोलते हैं ना आज चाय अच्छी नहीं बनी because they didn't have that exact SOP. वो सेम लेडी जब घर में बना रही होती है she knows I have to put these two spoons with this spoon only. This much of tea powder, this much of sugar, I have to boil at this much flame for this much time. Though it is not documented, but it is fit in her mind that how I have to prepare this tea. And if say some guest comes to my home, my wife is not at home at that time, and if I am in a hurry, I am preparing something, immediately that guest will say, "No, last time the tea was better as compared to today," because it didn't have that concept of reproducibility. And the same problem happens with our classical literature also. And this is not of today. I have just quoted one shlok from our classical reference from Ayurveda Prakash. It is already mentioned 300 years back. Pachit Shastra Kriya Nasti. Even our classical literature, at some places, the method of preparation is missing. If it is mentioned, how many rounds of that work are to be done, that is not mentioned. If that is mentioned, that form in which the half of things are mentioned and half of things are taken as granted, because in earlier days it was coming through Purushasya Parampara. That Guru was teaching it to their Shishya and then it was coming. But that lineage is broken now. So now we have different persons at different stakeholders at different levels. So we cannot go as such. So we are trying to prepare and establish a system through which we can get reproducibility. Whatever prepared by one person at one place using one set of materials will be exactly same as that by another person in entirely different scenario. For that reason, this AFI will help. So already we have many ingredients, number of drugs we are using. In some cases, even we could not till date, after so much of struggle, we are not able to identify that exactly what this material is to be called. हमारे पास कुछ आयुर्वेद के सिद्धा के यूनानी के टर्म्स तो है, but we don't know how what it is exactly. In case of plant materials also, there will be tremendous variations of opinion. One name is given in classical literature, one person will say, by this name I should use this plant. Then another person will say, no, no, not A, that B plant should be used. And again, C person will come, that A plant, but with little different species and or cultivated varieties should be used. So that is endless. And this confusion is less. So we add more confusion by using different nomenclature for same drug and same nomenclature for different drugs. So that is unfathomable. We cannot go to it. So, but as far as formula is concerned, we stick to one formula. We take literature from exactly one reference. We interpret it. So the word Amruta in Ayurveda, for example, it is used in case of Haritaki also, in case of Guduchi also. And even it is used in the case of Pasana, poisonous material also. So if you are following a formulation, this formulary will specify that in given case, if that word Amruta is mentioned, it may we are interpreting this Amruta as Guduchi with botanical identity as Tinospora cordifolia, and it same will be used as the part, not any part of that Tinospora cordifolia, with whatever one part each under that formulation. So this exact details we will give. If that material needs some purification processes, some shodhan, some musaffa, something, then that will also be included and specified in this formula. This becomes formulary specification for a given formulation. But here, like simple, we may mention in case of Ayurveda, simply say we are using cannabis, our word Vijaya. It will be mentioned in formulary. Vijaya Shuddha means that purified form is used. Same thing if it is the same drug is used in Siddha, we will mention Ganja, Suddhitadu. But what that Suddhitadu means, that we have to again refer to appendix given to that formulary. This being a regulatory document, we try to fit all the necessary information in one document. The attempt is that you should not need to refer any other document for any question arising out of that document. अगर कुछ पढ़ते वक्त आपको लग रहा है अरे इसका मीनिंग मुझे चाहिए है, तो मीनिंग उसी डॉक्यूमेंट के 2-4 पेजेस आगे पीछे में आपको मिलना चाहिए. With that, these appendices are also included, where all elaborate procedures and information of technical terms are given. In some cases, if that material is not available, we may permit some substitutes also. Classical books में कुछ mention किया हुआ था. Let's example, कस्तूरी, मस्त. अब वो लेने के लिए बोला हुआ है. वो तो 1 ग्राम देखने के लिए भी जो है वो पूरे भारत को सैंपल रखना है, बट वी आर कीपिंग 1 ग्राम ऑफ कस्तूरी एज ए म्यूजियम सैंपल, इवन दैट विल नॉट बी अवेलेबल just for visual look for entire India, where we will get kilos of kasturi to add to the formulation. Not at all possible to. So in such cases, we will give some substitute, or even in some cases, we will give omissions also. That though this material is mentioned as a part of ingredient, but in current circumstances, we are keeping that material. Another example I can give, why to go for air drugs only. Simple common example. Haridra Khanda, one Ayurvedic formulation. As per classical literature, it has to be processed by cooking it in milk. But if you cook it in milk, its shelf life is hardly 3 to 4 days. On fifth day, it will get spoiled, rancid with fungus, and the consumer will get vomiting. So we have made it change that the formulation will be prepared by using water only, and when the person is consuming, they will take it with milk, so that we ensure that it is having good shelf life and it is available in a healthy condition to each person. So in such way, we are developing our formulary and pharmacopoeia. So specific stages of preparation of formulation and its doses and the appendix also. I already discussed technical terminologies, the purification processes, different nomenclature clarification. All these things are also included in appendix and it becomes our formulary. Then we will come to pharmacopoeia. As already discussed in the first picture, we have shown a picture of part 1 pharmacopoeia where single drug ingredients are covered, and the second one is part 2 where formulations are. In API, the pharmacopoeial monographs precisely prescribe standards to ensure identity, purity, and strength. I repeat, identity, purity, and strength. Because in Indian scenario, in our drugs and cosmetics act, we don't need safety for our classified drug. If it is our drug, we don't need safety unless you are deviating from its given literature that you have already seen in Rule 158 in the day before yesterday's lecture also. That where we have to go for safety studies and where we don't need those studies. But in general, we don't need it. So identity, purity, and strength. These things will be basically covered. Botanical identification wherever required, various physicochemical parameters we are giving in assess for elemental assays or functional groups or some marker compounds wherever applicable, we are adding to our pharmacopoeia. And sophisticated analytical techniques, which we have already taken hands-on exposure for the SPTL, those are also used. So in case of part 1 pharmacopoeia, it becomes for single drugs. Here, most of the things becoming are kind of from plant origin. I have just discussed over the standard format for a monograph of plant origin. The monograph will start with title of monograph, Ashwagandha. The drug name will be given as the heading itself, and then there will be a definition. Ashwagandha consists of dried, mature root of Vidania somnifera. So Vidania somnifera, that botanical identity we gave the part used root. We gave it harvesting condition, dried, mature root. So that exact description, how that material is to be used, that is given here. But before entering to the flow of a monograph, I just want to clear two things. Pharmacopoeia is a document used meant for regulatory enforcement. It is a legal document. But that doesn't mean that each and every line, each and every word mentioned in the document is legal. So, for example, here we will describe. So, the first thing, whatever we saw, title of the monograph, Ashwagandha, that has regulatory purpose. Definition, you have to use dried mature root of Vidania somnifera. It has regulatory purpose. But again, we will use some classical names, whatever different synonyms given in our classical book or regional names mentioned in other languages. Hindi में ये कहते हैं, मराठी में ये कहते हैं, तमिल में ये कहते हैं, नेपाली में ये कहते हैं. If that description is given for that doesn't have a regulatory purpose. That is just informative thing. So the monograph consists of some information of regulatory nature and some information of informative nature. So that we will specify at every point. So here the initial title and definition are regulatory. Classical names, names in other regional or the English language or whatever vernacular names, those are of informative purpose. That they don't have any regulatory purpose. Then comes description. macroscopic features, microscopic features, its powder features, from the point of pharmacognosy we are giving. And whenever you are seeing that powder under microscope, whatever features are to be identified for its better understanding, self-explanatory diagrams are also given there. We are giving diagrams, means it is handmade sketch. We don't give photograph, because if we give photograph, that will become a binding to the regulator. ये जरूरी नहीं है कि अगर एक फोटोग्राफ में दिखाए हुए में एक अगर कोई स्पिंडल शेप फाइबर 3 मिलीमीटर का दिख रहा है, वो हर प्लांट में 3 मिलीमीटर का ही हो. So we are avoiding here photograph. We are giving schematic diagram. We just mention how it will look like. That is not exactly for the exact scale. So this information we are giving for regulatory purpose, but avoiding any conflict of natural variation. And here, but I will stress, whenever we talk of quality standards of any drug, any material, everybody of us, by default mechanism in our brain, we focus on the issues related to its chemical analysis. Botany always remains a neglected portion. Even a person who is himself or herself as a PhD in pharmacognosy, but when they take drug in hand, they will be more interested in how the TLC is looking, what reagent is used, and what markers have been identified. There is very less focus given on actual pharmacognostic parameters. If while visiting any industries for their inspections, etc., also for chemistry, if we ask what is your staff in chemistry section, they will make 10 people standing in the row, right from their deputy manager to working assistant. There will be 10 persons working under the exact quality control chemistry section. And if we ask, then what about pharmacognosy or botany? Then they will say, our general manager had botany in his graduation. Nothing else. Whether general manager is going to come and stand at the QC lab? No. But just they will fulfill that record by saying that yes, he had botany as one of his subjects, so that we fulfill our staff requirement. Nobody will focus on that parameter, neither for manpower nor for work for anything. But the difference is that the basics of analytical aspect, whatever we are having next parameter, like our hash value related to or even whatever assays also we are doing, they all those are of non-specific nature. But the parameters under pharmacognosy are of specific nature. What does it mean? Like water soluble extractive आपने इसका देख लिया, हमने फार्माकोपिया में लिखा हुआ है not less than 10% होना चाहिए. For example, whether if your sample fails to that given parameter, or even if it passes, चलो पास कर लिया मान लेते हैं, not less than 10% लिखा था, आपकी वैल्यू 11% आ गई, you say that yes, this parameter is fulfilled. But whether as an analyst, can you say ये अश्वगंधा है? Chemical parameters don't lead to a conclusion. They just say that this material is not bad. बुरा नहीं है, इतना बताने का काम केमिस्ट्री करती है. अच्छा है बताने का काम केमिस्ट्री नहीं कर सकती. Whatever our current knowledge. But botany does. The botanist with his powder microscopy identification will give a strong statement. Yes, this is root of ashwagandha. अच्छा है बताना है तो botanist चाहिए, बुरा नहीं है बताना है तो chemist. And this is always given a lesser importance, and we miss that botany and we miss those specific observations, and we just beat around the bush. हम हाथ में डंडा लेकर सांप छोड़कर इधर उधर पीटते रहते हैं केमिस्ट्री के पैरामीटर. So I urge everybody to keep this in mind for throughout your life and give your more attention as far as possible to their pharmacognostic identification. बॉटनी थोड़े हैप्पी हो जाएंगे, केमिस्ट दुखी हो जाएंगे. Sorry. Yes. बिल्कुल बिल्कुल both are required. See, it is a two-wheeled car. You cannot go with one wheel broken. But the thing happens, we only focus on chemistry. That's my That is the way excitement. Even he will do something, and whatever he does is the analysis at his level. That is the analysis. It is not the naked eye examination. Whatever is naked eye, what the teacher teaching is the analysis. The definition of analysis changes in that case. Analysis may not be done by statistical experiments only. So then we go to the next thing covered in our pharmacopoeial monograph, chromatographic identification, HPLC, HPLC, whatever, and other phytochemical reference markers also wherever possible we have mentioned. Now from here these 2 points, that whatever related to pharmacognostic features and this identification, these are mandatory, these are of regulatory nature, these are essentially to be complied when you are checking for it. Physicochemical parameters. Some list I have given which are used commonly: foreign matter, loss on drying, total ash, insoluble ash, sulfur soluble. These are mostly common use, and other insoluble additional that will depend on the nature of the material which we are analyzing. Accordingly, so this will change. Here, in each case, we will be giving some prescribed values. That what is permissible for loss on drying or in each of these. Those values are to be seen from 2 different perspectives. In some cases, we will be giving it in the form of range, and in some cases, we will give it in the form of limit. Range means outer end and lower end, both will be defined. For example, for pH, we will say pH has to be between 5.5 to 6.5. So the lower permissible limit and upper permissible limit, both things we have given as a range. So anyway, deviation will lead to failure in that quality. But in most of the cases here, we are giving limits. Means one side will be closed, other side will be open. Like in case of foreign matter, not more than upper limit is 6 up to 2%. But in case of like total ash, not more than 3%. Here upper limit is 6. Lower there is no limit. That can be anything. But in case of alcohol soluble extracted, it will be not less than 13%. So lower limit is 6. Upper that can go anywhere up to 100. So only one side exclusion criteria are given in some cases in form of limit, either from lower or from upper. And in some cases, exclusion is given from both sides. Here, specifically for persons giving analytical reports from regulatory purview, just use your common sense whenever you are finalizing the report. If that, say for alcohol soluble extractive, we are giving the same example if I take not less than 13%. And if your calculation comes as 12.9%, for God's sake, while preparing report, simply write it as 13% and close the matter. Because as a law, 12.9 is failure. Only 13.0 becomes as pass. But you know personally that won't make any difference for that minor fraction. So if it is just a round figure related thing, just please ignore those things and come to the acceptable range so that there will be no more headache for you. Then this became essential parameters. Then there are some other requirements. We are using the word other requirements because they won't change based on the nature of the drug. इससे पहले हम जो देख रहे थे उसमें ड्रग कैसी है उसके अकॉर्डिंगली पैरामीटर चेंज होने वाला. The next limit for heavy metals, whatever is the material, the limit for heavy metals will remain same. Whatever is given by WHO, we are adopting them. And exactly 4 metals we are taking here: mercury, lead, cadmium, and arsenic. Microbial contamination we are discussing. There will be total count, and again for specific pathogens. Already we have covered it under pesticide residue, that list of 24 pesticides we will be testing. For aflatoxins, B1 individually and B1, B2, B1, B2 collectively, that we will be testing. These limits won't change from material to material. That irrespective to material. Only one thing here to note is in case of heavy metal. Say mercury, lead, cadmium, and arsenic. We are telling these 4 material heavy metal limits are to be tested. For the sake of discussion, we will use just word heavy metal. arsenic as such is not heavy. If we strictly see the definition of chemistry, but let's say it as heavy metal. But the simple example, Ayurvedic formulation, Arogyavardhini. It has mercury as one of its ingredients. It contains almost 3 to 4% mercury. Whether this 5 ppm limit will be applicable in such conditions? Obviously no. But even in that case, other limits for lead, cadmium, and arsenic will apply. If any of this, cadmium is nowhere used in any of our systems, but mercury, lead, and arsenic may find place as a standard addition in the formulation composition itself. In such cases, if that material is used as a part of that medicine, then it will be exempted from the heavy limit testing. It won't apply blanketly for each. If it is a pure herbal material, obviously no question, all limits will apply. But in case where metal mineral based drugs are used, and if any of these is used as ingredient, not only as ingredient, it may be used as a intermittent in the procedure itself. Persons conversant with Ayurveda may know. Somanapti Tamravasma. Basically, it is a Tamravasma prepared from copper. But arsenic trioxide is used as a material to prepare that Vasma. So, in such case, where basic material where to start for that Tamravasma is copper only. That is not a heavy metal. But arsenic trioxide is used as a material in the process. So there also that arsenic will be exempted from that test. Then some assays are also used with their limits and quantitative estimations for permanent functional groups. They are also mentioned in some cases. And phytochemical reference standards are also mentioned in some cases. Here again, for all these tests, whatever we are performing, details will be given in the last appendix. But here is the word of caution. For pharmacognostic identification for HPTLC, here for those phytochemical reference standard mentioned, and here these physicochemical parameters. Wherever the tests where such physicochemical tests are mentioned, the test in cases where the result is dependent on the methodology used, there we have to use specific method mentioned in that pharmacopoeia only, like for loss on time. लॉस ऑन ड्राइंग के लिए आप अगर उसको 105 डिग्री पर करोगे या 100 पर करोगे या 200 पर करोगे, your output is going to differ. So in such cases, exactly the method mentioned in pharmacopoeia has to be adopted. The results are not true or false unless exact method is used. But if we go for the heavy metal estimation or quantitative assays, lastly mentioned, where the just last quantity matters and the testing protocol is irrespective of for the result. Whatever method you are using, whether you go by atomic absorption or you go by your exact mentioned hand-hold traditional methods or you use ICP-OES, that heavy metal content or mercury content elemental analysis, the result will be same, irrespective of method followed. So wherever methods and results are independent of each other, as an analyst, you are free to use any method. But where methods and results are dependent on each other, you are bound to use the method described in pharmacopoeia only. And even wherever, in case of that independent results also, if you are using different methods, you should ensure yourself that by both of these methods, the result is going to be at same level. There won't be any difference. If any person from Ayurveda background, they can understand easily. First year graduation, just you can remember. ESR by Western Green method and by that another method. If just method for ESR is changed, the result will be changing. Here also it may happen. So ensure yourself that the same method is giving the result exactly as per pharmacopoeia. And even if you change the method, that won't give you liberty for the parameters mentioned. मैंने दूसरा मेथड यूज किया था इसलिए दूसरा रिजल्ट लागू होगा, ये वाला रिजल्ट मेरे पे लागू है, ये नहीं। रिजल्ट आर मैंडेटरी, methods in case of independence that can be deviated. Then some information about its dose, properties, actions, formulation, usage, etc. is also given. This is a non-regulatory informative information given just for the sake of knowledge and for better understanding of the person using it. Similarly, for formulation monograph, here also more or less all the things are same. There also there will be a title, definition, formulation, composition exactly as given in the formulary. And method of preparation, here we are giving stepwise method of preparation. Here also this method of preparation is indicated. Like this you can go. Method of preparation can be deviated provided there is no variation in result for next parameter. Identification is done by powder microscopy, but powder microscopy we restrict it up to 10 ingredients in situ. If that is material is cooked, cellular structures are lost. च्यवनप्राश बनाते वक्त आप उसमें जो है आमले का फार्माकोग्नोसिस नहीं कर सकते because cellular structures are lost. But if it is a just powder formulation by adding some different kinds of powder material, then cellular characters are not lost. We can verify them. But in many cases, those will be overlapping with each other. So we restrict ourselves to 10 ingredients. If the number of ingredients is more than 10, powder microscopy is not done. Then comes chromatography, then physicochemical parameters, same as we discussed in case of single drug. Then assays, limits, these all things are also exactly same as that. Now it comes to whatever other things other than monograph we are covering under our pharmacology. Firstly, general knowledge. This specifically insisting because if you are reading a pharmacopoeia and if you need interpretation of any statement made in pharmacopoeia, take it in the context of entire pharmacopoeia from its initial to last. Only reading one line may give you a misleading interpretation. Monograph में लिखा होगा ये यूज करनी है, but if it in general notices it is mentioned that it is exempted, then that line has to be read in both contexts simultaneously. So general notices will give guidance for manufacturers and analysts. Permissible deviations from composition, processes, substitute, deletion, addition of or anything or any banned items related excuses. So whatever preservatives can be added, whatever changes in dosage form or methods can be done, those all things will be described in general notice. At last, appendices are given where detailed procedures are given. Which procedure is to be followed word to word and which is to be taken as indication we have already discussed. Stability testing is already this is taken care by this is just illustration of how we have given space analysis standards in our latest version. This is agreement illustration for problem one the details are given. Then yes, apart from all these pharmacopoeias, we have some instances we have prepared our atlas documents also on PhD and macroscopic atlas. These atlas documents are just as an additional tool for analysis and they don't stand as a regulatory document. Violation of atlas will not be treated as an offense. It is just informative document, completely non-regulatory. These are documents of atlas on PhD and this is on the microscopic atlas used here because this is a non-regulatory document, so pictures are given for exact understanding of the similarly in case of chemistry also all those details are available. Whatever we saw for Ayurveda is exactly the same in case of Siddharth with the pharmacopoeia of India and the formulary of India. In case of Unani also only small changes. In case of Unani the formulary is called as national formulary for Unani medicine. And in case of homeopathy, all other things will remain same for pharmacopoeia. But homeopathy as a science itself doesn't recognize formulation. There is no concept of formulation in homeopathy. So the homeopathic pharmacopoeia goes only for whatever we have part 1 in our pharmacopoeia. So only one series of pharmacopoeia is there. No R2 series and no formulary in case of homeopathy. This is just statistical information about total number of standards till date. These are figures of our different these are our youth path related different initiatives where we deviated from all this structure and went for solitary standards for Wikipedia as well as for apart from all this. So नहीं बजट का हेड भी है Yes. ये सब है चलो पता ही एलएनटी से गया है कि ऐसे तो एक क्वेश्चन भी हाँ ये मैंने वो बजट हेड बता दिया अब वो बजट हेड कहाँ से प्रोसेस होता है वो तो मुझे पता नहीं। ये बजट हेड मोस्ट पाँच तीन चौबीस का मतलब जो है फ्रेश क्वेश्चन वही आपको बोला जैसे ट्रांसलेशन वगैरह करना की पूरी प्रोसेस कैसे करनी है उसके बाद उसको वेबसाइट पर जी Apart from all these pharmacopoeias, recently we have launched one initiative, one OH, one herbal standard. So here we are trying to bring all the standards under one document. Regulatory provisions for this are under consideration. Very soon you will soon that was from my side. Yes, yes. Obviously, there are notifications from Ministry. It is already planned. Even in pharmacopoeia also, you will get this information in general notices, and Ministry has issued separate notifications on this. Anything else?
Sir, in the research studies, there will be like different companies may have different level of mercury in the formulation. So, is there any fixed standard telling that this formulation should have only this much of mercury or
That
content?
depends on the reference.
Yeah, but the outcome of a formulation might, whenever you analyze, it might show different variation than the
Because if reference changes, that you cannot control it for
correct,
the
yeah, exactly. So
in
it may,
given reference, it should be there. That to wherever we have given our standards, then there will be mandatory. But if our standards are not available, then in-house standards are to be used by the manufacturer. That way, if it is a proprietary medicine, there won't be any standards from government.
okay, so then it becomes difficult to control the immune toxicity profile and everything.
Yeah, that variation will be met, but as of now, there is no answer. Okay, thank you all. I hand over mic for next.
Thanks, Dr. Nikhil, for his so informative lecture on the pharmacopoeia and formulary specifications of Ayurveda, Siddha, Unani, and homeopathic drugs. Now we have our next speaker. He is Dr. Mukesh Kumar. He is the HOD, Pharmacology Institute of Teaching and Research in Ayurveda, Jamnagar, Gujarat. He will deliver his lecture on the safety evaluation of Ayush drug and experimental scientific approach. So I would request Dr. S.C. Verma sir to felicitate him with the flower bouquet. Sir has completed his M Pharma and PhD Pharmacology and working as a head of pharmacology at ITRA Jamnagar. He has more than 20-year experience in the teaching and research. He has received Young Scientist Award in Drug Research by CCRAS, Ministry of Ayush. He has guided more than 24 M Pharma students and 70 PhD research scholars. He has worked as in charge of the Institute CCRAS at Ahmedabad, coordinator of Department of Project, Gujarat Ayurveda University, member Ayurvedic Pharmacopoeia Commission PCMNH, coordinator of Technical and Research Cell of IPGT and RA. So I would request Dr. Mukesh sir kindly deliver his lecture.
Very good morning to all. I'm basically associated with the preclinical safety and efficacy of Ayurveda. So I mainly my close concern with the reverse pharmacology concept, how the this modern concept quality Ayurveda in terms of safety and efficacy. So safety evaluation of Ayush drug and experimental scientific approach. So you all of you know the drug discovery program, that is conventional drug discovery program, starts from target identification to preclinical and then clinical. During this conventional discovery program, most of drugs enter to the market, and you see the scenario since last almost I took from 1997. So very few drugs approved worldwide for new chemical entity. You see the surge in mainly biological approved by means of FDA for the drug regulatory aspect. But you see, this is very low. Why very low? Worldwide, 2023 is very low, and it's very risky, and the success rate is below 1%. Huge investment and also time duration is very big for the new drug discovery program, almost 10 to 12 years. That's why most of times consumed by means of clinical trials. So before clinical trial, there is preclinical trials. In preclinical trials, not always the pharmacology. Preclinical trials include all the drug development as well as the preclinical trials. And you will be very surprised when drug moves from preclinical to clinical, almost 90% drug fails. Why drug fails? Because previously there is experiments on the animals, but when drug moves preclinical to clinical, there is chances that the same results may not obtain when drug moves from preclinical to clinical. That's why 90% drug fails. It doesn't mean that there is no requirement of preclinical trials in case of traditional system of medicine. Even you see, when drug move from phase 1, phase 2, phase 3, there is chances that drug fails. Even drugs approved in the market, then also there is chances of the drug failure. This is the scenario where the drugs fail in phase 1, phase 2, phase 3, and even after approval to the market. You see, the most of reason of drug failure during phase one is of safety, because you know the phase 1 trial mainly deal with the safety and efficacy in healthy volunteers. Phase 2 in disease person, phase 3 multi-center clinical trial, and phase 4 is post-market surveillance. So most of chances that the drug failure at its safe, the percentage may vary, but there is chances. So even preclinical and clinical, all are predictive models. When drugs approved for that marketing, then there is also chances. You see, worldwide withdrawal since 40 years, the most of drugs fail, that is of hepatic toxicity. Why hepatic toxicity? Most of drugs, because these are the first cited organs. First cited organ include GI tract, liver, kidney. These all are first. The drug move through absorption, that is of kinetic profile, from GI tract to liver, that is by means of metabolism, and excretion through kidney. So these are
first cited organ, so there is chances that most of preclinical safety issues concerned with the hepatic. So major reason of again that drug failure and the drug issue, that is of drug interaction. Drug interaction, why drug interaction? Because of the increase in frequency of polypharmacy. Most of the people, even the physician, they are prescribing drug together. For one disease, they prescribe a lot of drugs. So there is chances that the chances of drug interaction and drug interaction always result into drug safety. There is issue of concern of the drug safety. And all of you know, we have the parameters for the evaluation of drug, standardization of drug, that is a single formulation. But we don't bother about the drug interaction, particularly in biological system. So drug interaction is major part is of concern. You have to evaluate those safety and efficacy in case of biological system. And again, one more concept, that is a polypill concept. What is polypill concept? Nowadays it's boomerang. Polypill concept particularly prescribed for cardiac disorders and many more disorders, where the multiple of drugs required for the single disease. I explained the example, the polypill, that is metformin, aspirin, statin, and inhibitor. You see, all drugs make together. So one is for hypertension, one is for diabetic, one is for anti-hyperlipidemics, and one is for that is different indication of drugs. They are put together. But what are the issues? When you prescribe those drugs in a single formulation, there is chances that the interaction among the drugs, either chemical interaction or either biological interaction. And this is in case of modern drugs. But when you think of the traditional system of medicine, either Ayurveda, either Unani, homeopathy, there is multiple of drugs. We never bother about the mixing of drugs. There always to be concerned when you mix the drugs in a one formulation. And you see, during COVID time, the people are used to make the combination of Patyadi, Dasmul, and the Kuruchi. No, nobody bother about what are the composition, how it reacts when you put together, and how to evaluate those drugs, whether really they put the single ingredients or not. So there is always concern when you mixing those drugs, you have to prescribe the evaluation parameters, either for the chemical part or either for the biological part. You see, I put the one example, that is of glycosterol. You see, many ingredients they put together, and on that side I put the another example. They also put examples. You see, when you think of the traditional system of medicine, the drug acts as a whole, holistic approach. But when you extract something, it is not holistic. For example, opium is acts as a whole, but when you extract morphine is pure compound. Same way, when you extract antioxidant, when you extract, for example, when you extract vaccine or vaccinol from Vasata, it's not a Ayurveda or natural, but people sold in name of natural. They are mixing many of this extract together and sold as a herbal or sold as a, for example, this is natural. No, it's not natural because it's possibility that those compounds, when you extract as a pure compound and mix together and give to the biological system, it may produce serious adverse effects. I will give you example. Recently we conducted the work for the master degree scholar, the Holar that drug, that Aconitum heterophyllum. That Bal Chaturbhadra Guna consists of Aconitum heterophyllum. But what the chemistry student, so what he did, he done the extraction. Actually, he planned for the antipyretic study. So he done the extraction, then successive extraction, then it came to be. He started the work with 50 milligrams. Just giving lines of 50 milligrams, immediate mortality was observed. Why mortality? Because when he extracted that successive, I think maybe aconitin or something other the constituent may get extracted from that part and produce the serious toxic effect. So that's why when you extract, that's why garlic extract, phytosterol, gamma, so they put together without evaluation and that market, this product also available on Amazon. But what is safety, what is the efficacy, we don't know. So that's why polypill concept, that the even pharmacopy also has to develop the chemical markers, so you have to develop the evaluation parameters for those combinations. So again, one more study. This is from modern side. You see, modern people even not allowed the more than 3 to 4 ingredients together. So they even many times they ban those formulations which consist of the combination. You see, the combination of these antibiotics, even two and ofloxacin widely prescribed in India for GI tract infection, and many drugs which are in combination. Most of combination observed in cold and cough type of disorder. You see, and CDC also routinely they are banned those formulations which are in combination. And you see in the rational system of medicine, without any rational, they are adding those things. So the reverse pharmacology concept is must for traditional system of medicine. Reverse pharmacology concept for modern medicine, they have to extract something, they go for sustained efficacy and prescribe it. But for traditional system of medicine, whatever the red, the scope is to understand mechanism of action at multiple levels. The scope is to understand the mechanism of action. Mechanism of action is must. Drug at what level, at which dose, at what time, and at what time excreted from body. And another part to optimize safety, efficacy, and acceptability of drug. Here I miss one more part, to develop the chemical markers and biological markers. Because you see, biological marker may be chemical marker, but chemical marker not always to be biological marker. Chemical marker when moved to the biological system, it may change due to metabolism, due to some other parts, due to interaction, we don't know. So that's why you have to prescribe those chemical markers and biological markers and evaluation parameters for those markers, how to standardize the drug. So this already I think you studied for this reverse pharmacology. This was prescribed previously by India. You see, survey of traditional remedies, choice of plant identification. So these parts from clinic to laboratory. So conventional drug discovery program from laboratory to clinic, but here clinic to laboratory and laboratory to again clinic. Modernized technology, new dosage form, again go for the clinical trial and commercial production. Okay. So here you see but again, the major part required, that is of pharmacology, toxicology, clinical pharmacology, pharmacodynamic and biology. And most of the traditional system with those parts. And no one drug has the full profile which can deliberate what is this safety, what is this efficacy, what is bioavailability. Bioavailability is not possible as a whole drug when you think of the holistic approach, because it is combination of phytoconstituents. That's why bioavailability part may not be that much helpful to the traditional system. You see, that's why the whole holistic, when you think of the traditional system, you always prefer the holistic approach. And without holistic approach, you can't go for the with the single study. You cannot claim those parts. If, for example, you're doing simply in silico and you tell that this drug may be useful, no, it's not possible. Why? Because it consists of polypharmacology, it consists of many ingredients, it consists of many phytoconstituents. Which constituents is responsible, we don't know. That's why you always start from in silico, computational methods, in vitro assay, cell-based assay, preclinical trials, and clinical trials. And at last, after clinical trial, you should always float the network pharmacology, that is of polypharmacology. So best word is polypharmacology in place of pharmacology for this traditional system of medicine. So you see some bioinformatics tool that is of SVCDME, Lipinski rule of 5. I think you all may know this bioinformatics tool. So we always use it. Almost 12 PhD scholars passed under my directive. So we always prefer the holistic approach for traditional system. If you go for the extract something and reporting something, then again the Ayurveda people or Unani people or Homeopathy people, they always ask you what is the importance to the traditional system, because you are doing the work on extract, and extract may not directly correlate with the Ayurveda, with the Unani Homeopathy, because the single extract we never use in our practice. Mostly the water extract is using, but other extracts we are not using. That's why when you go for the such evaluation, at least you have to extrapolate all these studies, then only you get something to find out the probable mechanism, to find out the probable safety and the efficacy. Lipinski rule of 5, Protox 2, this predicts the oral toxicity, hepatotoxicity, carcinogenicity, then tox free. These all are available free online. So these are certain in vitro cell lines. This is sensor cell lines, normal sensor cell lines, liver kidney cell lines. Always FG2 and chain liver cells, most widely used cell lines for checking the toxicity of drugs in vitro. But here, the major issue with the metal and mineral preparation. You have to know the Bhasma preparation, widely used in Ayurveda and other system of medicine also. So what is the issue? When you put this type of preparation with the cell line, soluble, then only it may come to contact with the cell. And people always write the drugs at the dose level of 100 mg per kg did not produce any toxicity. So this statement is right? No. Why not possible? You have to prescribe which cell line, at what dose level, what is solubility. If it is not soluble, drug remain in this part, cell remaining in this part, it will not get interacted. If they not get interacted, how drug produce the effect when you claim on the safety? So better to, that's why I told you, better to correlate those data always with the in vivo study. Because in vivo study in which you correlate, then you get something. Without in vivo study, you can't claim. So I put some cell lines widely used for checking the toxicity, safety, and efficacy. Same way, neuronal cell lines, neurotoxicity, immune cell lines, these all are cell lines. So routinely used for the drug safety assessment. And we widely use some cell lines, for example, Dengiver and some cell lines for wound healing activity, then cardioprotective activity, then kidney protective activity. Routinely, we are using those cell lines. And sometimes when you use the herbal extract, we get very good result. But when you use the Bhasma preparation, you have to be very careful for the dilution of drugs. Because why dilution of drugs? You see, when you put herbal drugs in water, you put the water extract, okay, then serial dilution from 100 to 50, 50 to 25, 25 to 12.5, like that, serial dilution. But now, when you put Bhasma in 10 milligram per 10 ml, you put dilution 110, 1 ml you extract from pipette from this, and when you transfer, how you get finalize that 5 ml consists of 50% of Bhasma, because it's not in solution form, it's in suspension form. So that's why go for serial dilution, exact serial dilution not possible in case of if drug not soluble. Okay, and probably this happen in case of Bhasma preparation, after the metal mineral preparation, turbo mineral preparation. So that's why it will be very careful. And why this serial dilution required? When you go for the cell line study, always dose is 100 microgram or 10 microgram. 10 microgram is not possible to weigh, weighing machine. So that's why they prepare the preparation 1 milligram per ml, 10 milligram per 10 ml, and then go for serial dilution. So be very careful while doing the in vitro study in case of herbal preparation. This is the extract, some of the extraction technique. I will not go in detail for this part because already chemistry people explain you very well. And another reason, that is of major reason, lack of Ayurvedic knowledge for mixing of drugs, as already I explained. Now industry people, most of they are doing the mixing of drugs without any rationale, Simply they understand it, how to prepare adaptogenic drugs. They simply use the Ashwagandha, Brahmi, and they are mixing and so on. No any regulatory guidelines, no any specific strict guidelines or strict evaluation methods to stop that to mixing of those drugs. And there is even many DIs and that from the regulatory agency. There is no guidelines prevailing in India. So that's why the people are using the mixing of drugs without any risk. Already I given the examples during the COVID time. Many formulations came in the market within 5 to 6 months. Many formulations without any risk. Same way, multiple phytoconstituents. Third one, selection of incorrect Anupan and Sulfan. Very important aspect when you think of the traditional system of medicine. Most of traditional system of medicine always prescribe with Anupan and Sulfan. Anupan, mostly honey, ghee, honey and ghee mostly widely used, but other Anupan also there. Lot of Anupans they prescribe for the administration of drugs. So sometimes if you are not using the Anupan or Sulfan concept, the drugs may produce toxicity. I'll give you one example. We started the work with Tamrapatna. We've done lot of studies. I'll give you this example. Why? Because here we observe the exact role of Anupan. When we started the work with Tamrapatna, student came and administered to animal at the dose level of TD, TD5, and TD10. The death observed, mortality observed in animal. Then we think, what is the reason of death? Then we search and prepared, review the protocols. In last we found, actually when he administered, simply mix the Tamrapatna with water and administered directly to the GI tract, directly to the stomach. Because when you administer to animals, the catheter we use, the catheter bypass the mouth. directly administered to the stomach. What happens? It produces the toxic of the Tamrabhasma in GI tract. So again we reviewed the protocol. Actual method, Tamrabhasma, first you put it in honey, make the paste, and by means of licking method. So when we administer through licking method, nothing was happen. So there are the concept of Anupan and Saipan, and you should always mention those Saipan and Anupan. And those part we missing in the analytical or chemical part, because you have no any such parameters of the evaluation when you mixing drugs with Anupan and Saipan, how to evaluate those drugs, how to standardize those drugs. Another adulteration, substitution, wrong drug. I give the example at last. What will happen when drug wrongly misinterpreted? Same way, therapeutic incompatibility. When you put the drugs together, maybe chances of therapeutic incompatibility. The Rash Guna Virya Vipak concept. Rash Guna Virya Vipak. The concept, when you don't bother about those concepts, when you put the holy drugs together, maybe chances that you may not get the effect. Limited proven resources, that is of preclinical and clinical. No drugs have foolproof monographs on preclinical and clinical. So what is the reason of safety? When you give drugs and drugs may accumulate, that is uptake is more than elimination. K1 is more than K2. So accumulation of drugs, and drugs, if it is not excreted in proper manner, then drugs may accumulation and produce the safety concern. So administered now previously all of you know the pharmacology and toxicology. Nowadays toxicology is separate branch than the pharmacology. And even in toxicology also toxicokinetics, toxicodynamics, toxicogenomics, many branches are now evolved. And you should always be concerned when you talk about the safety of drugs, you should also concern about those toxicity guidelines and toxicity profiles for the evaluation. So ADME profile, same as pharmacodynamic, pharmacokinetic, same as toxicokinetic, toxicodynamic. So safety aspects, particularly in concern to the reverse pharmacology. Why? Because use of processed metal, minerals, mercurial, herbomineral formulation are reversed, particularly Schedule E1 drugs. Schedule E1 drugs, all the poisonous drugs in the traditional system of medicine, that those given in Schedule E1. Unique procedure. So the Maran, Amritikaran, nobody bother about this procedure. Because when you process for the Sodan or Maran or Amritikaran, what happens at each stage? What happens even final drugs prepared? So you have to be very foolproof plan for the evaluation of those drugs. Okay. Same way, role of Anupan already I explained. Most of methods are very tedious and lengthy. You see, Nag Bhasma, I think all of you know Nag Bhasma, the Bhasma prepared from the lead. It requires 60 foot, 100 foot. So whether the industry people pass through all these tedious procedure or not, or directly they prepared and put it in the market. So the specific testing protocol should be there. If you don't have the testing protocol, how you decide whether they process for 30 times, whether they process for 60 times? So another again JAMA articles, all of you know JAMA, Journal of American Medical Association, in 2006 to 2009 time, they reported for heavy metals content in Ayurveda. So many drugs banned in the most of European countries due to heavy metal concern. For the toxicity, you require large amount of compound. Why? Because when you administer drugs at different dose level, therapeutic dose level, therapeutic 5 times dose level, therapeutic 10 times dose level. If your therapeutic level is, for example, 1 gram, animal dose is, for example, 90 milligram, 5 times 450, 10 times 900, and that to be administered for 90 days, 120 days. Now you think how much drug is required for the safety trial. The purity of compound needs to be very high. If it is drug is not pure, then there is chances that the impurity or some other parts may also produce the safety concern. And you think of that drug may have the toxic effect. If additive excipients are not safe, then they become part of safety assessment. And this is again major issue in traditional system. Why? People are now told that the drug is natural, Ayurveda. But when you read the label, only 2%, 3% is the extract of Ayurveda. And other 97% that is from modern excipients. For example, any cream, facial cream, any cosmetics, anything you can take it. Okay. The herbal parts mainly 5%, 10%. Other part all are additive, excipient. That is of the modern concept. That designed for the modern concern. So you have also evaluate those excipients or those additives also as part of safety concern. And toxicity profile need to be documented in IND. IND is very important for an investigational nucleus. You know three types of IND, major two class of IND. One is commercial, another one is non-commercial. And under non-commercial, that is research IND, investigator IND, and disease IND. So we almost all the person engaged in all the academic institution or government institute that all are deal with the non-commercial IND. But when you work in the company, you deal with the commercial IND. And commercial IND always under stringent criteria of the drug regulatory authority, because they have earn the money from those IND. But for the academic and for the other, they not have the commercial interest. That's why they get the easy permission for the IND. So dose conversion, when you have the for traditional system of medicine, you know the drugs already prescribed in classical books. So it's very easy for pharmacologists or toxicologists to convert those doses into the animal testing protocols. This is therapeutic dose. You just convert into the animal dose and administered for the safety trial. So this is an example. Just I'll give you this plant is Opuntia elatior. These are called as Hatlathor and very well known for its hematinic activity. And this project actually started by Professor R.N. Acharya sir, and I'm associated with him for preclinical trials. He took the work of clinical trials. I took the work of preclinical trials, and many works. Almost 5 years we've done the works on this, and recently we completed reproductive and developmental safety evaluation. Why reproductive? Because you see in Saurashtra region or Gujarat region, most of people consume this red fruit as a nutrition or as a hematinic purpose, and even those parts also prescribed to the pregnant woman. Pregnant woman daily 2 fruits and very tasty. Even we also take routinely. It's very tasty. But the major you not remove it correctly, then maybe chances that your tongue and that will damage. So properly you have to remove, and pulp you can consume. So this example just I'll show you. You see the evolution of hematinic, then hematinic activity, then reproductive, toxicology, and even clinical trials with the different formulation. Different formulation, matlab, we prepared it saule, we prepared its juice form, we prepared its extract, and many more formulations prepared from those and tested for clinical trial. And full profile of chemical, how to evaluate, what are the difference, and we found the betulin content, betulin content is mainly responsible for its hematinic activity. So again, this actually the sophisticated technique may be useful to the traditional system of medicine for its evaluation. Why? Because you see, when you give Triphala to so if you take something and administer, so and observe from outside by means of this sophisticated technique. So drugs at which time reach to which organ, you can target from the upper side. You see, just I'll give the example, bioluminescence of the rat. Just but again, all techniques are very costly. Single instrument itself almost 3 crore, 4 crore, or 5 crore. But anyhow, this is the future of traditional. For the exact evolution, exact movement of those drugs in case of biological system, you can find out. So what is its use? In vivo drug action. When drug reach to the target site, what is the dose target site occupancy? Pharmacokinetic profile, ADMET profile, biodistribution, uptake of drug, its clearance, receptor occupancy, whether drug reach to particular sites or particular receptor. If it reach to particular site, when drug cross the drug membranes, or what are alteration inside the drug. This only possible by means of these all sophisticated techniques, PK/PD relationship, and those all preclinical and previous parts support the clinical trial. And ultimately, it's validation of response. And you see, validation of response is only possible in human and through clinical trial. So clinical trial always occupied the prime importance in case of drug discovery program. And almost 60 to 70% of budget, required for clinical trial. see, new drug development costs 1,000 crore, 1,200 crores, almost 600 to 700 crores fund required for clinical trials. So that much of sophisticated parameters. So ultimate, what is your ultimate goal? Is to produce the efficacy in clinical trials. So validation is also to be possible in clinical trials. Developing and assessing innovative and traditional therapy. Now we come to the actual guidelines. The guidelines published by the that DG and CDCSO. You see the new drug and clinical trial rule 2019. Okay, so this guideline says toxicities should be comply with the norms of GLP. एंड मजे की बात है कि इंडिया में जीएलपी बहुत कम है इंडस्ट्री को छोड़ के, पर्टिकुलरली ट्रेडिशनल सिस्टम ऑफ मेडिसिन में तो कहीं पे भी नहीं है, बहुत कम लोग ट्राई कर रहे हैं। आयुष का भी अगर आप देखो जीएलपी लैब किसी के पास नहीं, सीएसआईआर आईसीएमआर के पास है कुछ एक जीएलपी। तो जब भी व्हेन यू थिंक ऑफ द ड्रग्स टू बी एक्सपोर्ट यू शुड ऑलवेज कंसर्न फॉर इट जीएलपी। ये अपना जो ये अभी रिसेंटली गाइडलाइन आया ना सीडीसीएसओ का, उन्होंने ये बता के रखा है टेस्ट सब्सटेंस एंड टेस्ट सब्सटेंस इन विट्रो एंड इन विवो प्रॉपर्ली कैरेक्टराइज्ड एंड स्टैंडर्डाइज्ड। ये बहुत जरूरी है। जो भी हम काम करते हैं सबसे पहले, इसीलिए जो भी केमिस्ट्री से जुड़े मोस्टली यहाँ जो लोग आए मोस्टली केमिस्ट्री एंड ड्रग इंस्पेक्टर सब अलग अलग फील्ड से है। तो व्हेन यू थिंक फॉर द इन विट्रो इन विवो या व्हेन यू थिंक फॉर द इट्स efficacy or safety concern, this part is very important, should be properly characterized, standardized। तो जब भी आप सेफ्टी कंसर्न, सेफ्टी के लिए भी मैं पेपर भेजता हूँ ना, तो अच्छे से जर्नल सबसे पहले पार्ट ये पूछते हैं कि भाई आपने सेफ्टी ट्रायल तो कर लिया, बट जो ड्रग आपने एनिमल्स को दिया वो कैरेक्टराइज था, सही में वो स्टैंडर्डाइज था, वो सबसे पहला क्वेश्चन उनका ये होता है। और कभी भी सिंगल सेफ्टी भेजते हैं ना तो कई बार एक्सेप्ट भी नहीं करते। आपके पास पूरा डेटा होना चाहिए, हाउ प्रिपेयर्ड, what are the source of drugs, what is the SOP of drugs for the preparation, what is this characterization part, how you characterize, and at last what is the protocol for safety trial। all documents, that is approved protocol, raw data, draft report, final report, histology, at least you have to store for 5 years। जैसे NADL GLP में भी ऐसा दिखा गया। यहाँ NDC का जो अभी नया गाइडलाइन है उसमें बोला गया मिनिमम 5 years तक स्टोर करना है। वेट आर्चर्स और ड्राई आर्चर्स बोलते हैं, शायद आप सबको पता होगा कि मोस्टली ये करना होता है कि ताकि जब भी उसको रिवैलिडेट या रिवैल्यूट करना चाहे तो कर सकते हैं। टॉक्सिकोकाइनेटिक स्टडी शुड बी इंटीग्रल पार्ट ऑफ स्टडी। जो मैंने बताया टॉक्सिकोकाइनेटिक आजकल बहुत इंपॉर्टेंट पार्ट है। उसमें मेनली ये 3 स्टडी है, सिंगल डोज टॉक्सिसिटी, डोज रेंजिंग स्टडी, रिपीटेड डोज सिस्टमेटिक स्टडी। डोज रेंजिंग अगर जब आप ट्रेडिशनल की बात करेंगे ना तो डोज रेंजिंग की जरूरत नहीं है। व्हाई? बिकॉज ऑलरेडी डोज इज ऑलरेडी प्रिस्क्राइब्ड। व्हेन यू थिंक ऑफ द न्यू ड्रग, डोज रेंजिंग स्टडी यू शुड गो फॉर। डोज रेंजिंग means what dose level drugs have the toxic dose, okay, and repeated dose toxicity study, that is 14, 28, 90 and 180 days। so this type of study they are prescribed। so again you come over here, while I'll put it later, you see the repeated dose toxicity study required for new drug as per clause 2 second schedule, where the data, ये पढ़िए, where the data यहाँ पे जो है ना animal toxicity as per special clause 2 is submitted and same considered by regulatory for country which is eligible to drug shall not be required to conduct in India। अगर किसी ड्रग का ऑलरेडी टॉक्सिसिटी स्टडी कहीं पे भी रिपोर्ट हुआ है तो फिर से करने की जरूरत नहीं है। लेकिन ये पार्ट आगे इस साइड वाला पार्ट पढ़िए, ये बहुत इंपॉर्टेंट पार्ट है कि एनिमल टॉक्सिसिटी, न्यू क्लेम्स, डोजेस फॉर्म, रूट ऑफ एडमिनिस्ट्रेशन, कुछ भी आप चेंज करते हैं तो अगेन टॉक्सिसिटी स्टडी इज रिक्वायर्ड। एंड मोस्ट ऑफ केसेस में लोग ये चेंज करते हैं क्योंकि क्लासिकल तो आज कोई बना ही नहीं रहा है। मोस्ट ऑफ फार्मास्यूटिकल में कोई भी क्लासिकल ड्रग नहीं बना रहा। ओके, तो क्लासिकल ड्रग नहीं बना रहा है तो उसको चेंज करने के लिए आपको सेफ्टी ट्रायल करना बहुत जरूरी है। ओके, ये अभी नया गाइडलाइन NDCTR, बट द मेजर पार्ट, ये अभी रिसेंटली डिस्कशन हुआ CCRS में, आचार्य सर ने इसका मीटिंग किया था कि रियली इसको डायरेक्टली अडॉप्ट करना चाहिए हमें ट्रेडिशनल को। तो उसमें आई एम आल्सो मेंबर ऑफ दोस कमिटी एट दैट टाइम। and long discussion was going on. Here the major issue is number of animals. Number of animals is one rodent 6 to 10, 14 से 28 दिन. लेकिन जब 90 दिन की बात करते हैं ना, 90 और 120 दिन का स्टडी करना है, तो नंबर ऑफ एनिमल्स कितने, 15 से 30 per set per group. Now you think, when you doing the safety trials, control, vehicle control, therapeutic equivalent, TD5, TD10, and minimum 3 dose level. Now you think one recovery. So total 6 group. 6 group per 15 से 30 एक सेट से. तो मतलब 2 सेट लेना है तो कितना हुआ, अप्रोक्सीमेटली 30 से 60 एनिमल. नाउ यू थिंक ऑफ 5 ग्रुप, कितने एनिमल हुए, 200 से 300 एनिमल. डेली 200 से 300 एनिमल को दवाई पिलाना. नाउ यू थिंक द कॉस्ट ऑफ दैट स्टडी, बिकॉज टॉक्सिसिटी स्टडी है ना, 1.5-2 लाख में भी होता है, टॉक्सिसिटी स्टडी 90 लाख में भी होता है. क्योंकि आप कौन से पैरामीटर को इंक्लूड करते हैं वो जरूरी है. कि टॉक्सिसिटी स्टडी में ऐसा बोला गया है, एज मेनी एज पैरामीटर यू हैव टू इंक्लूड इन टॉक्सिसिटी स्टडी. एज मेनी एज पैरामीटर. क्योंकि बाई चांस मान लीजिए आपने थायराइड पे स्टडी नहीं किया और बाद में पता चला ड्रग है, थायराइड को ही टॉक्सिक था, तो टॉक्सिसिटी में आप एक भी पैरामीटर को छोड़ नहीं सकते. सेफ्टी ट्रायल्स के लिए हर एक पैरामीटर को आपको कवर अप करना पड़ेगा. रिसेंटली आपको जानकर आश्चर्य होगा, अभी मेरे पास एक प्रोजेक्ट चल रहा है. ड्रग लिया है हमने आनंद भैरव रस, बहुत अच्छा ड्रग था आनंद भैरव रस, खास करके पायरेक्सिया में दिया जाता है. ओके, उसका मैंने डिटेल जीनोटॉक्सिसिटी ट्रायल, सेफ्टी ट्रायल, ओरल टॉक्सिसिटी और बाकी सारे पैरामीटर्स लिए एफिकेसी ट्रायल. और उसमें मैक्सिमम पैरामीटर्स भी इंक्लूड है, इवन हेवी मेटल्स इन सीरम, ब्रेन, किडनी, लिवर. एंड सबसे बड़ी बात उसका मैंने जीन एक्सप्रेशन, मिनिमम 10 से 12 जीन हमने लिए कि वेदर ड्रग प्रोड्यूस एनी जीन चेंजेस और नॉट. एंड on other hand, genotoxicity also be included. Genotoxicity, micronuclease से, chromosomal aberration से, form abnormality, वो हर एक पार्ट को कवर अप किया. तो टॉक्सिसिटी ट्रायल्स के लिए आपको हमेशा ध्यान रखना है कि टॉक्सिसिटी ट्रायल में एज मेनी एज पैरामीटर्स आपको इंक्लूड करना पड़ेगा. सिर्फ कोई 4 पैरामीटर लेके आप बोले कि मैंने सेफ्टी कर लिया, तो वो अगेन क्वेश्चन मार्क आ जाए. और मोस्ट ऑफ केस में आज लोग यही कर रहे हैं. जिसको ड्रग के लिए अप्रूवल चाहिए, अब डीआई जो है उसको एक्सपीरियंस होगा कि कई बार वो सीधा आपके पास क्लेम सबमिट कर देते हैं कि मेरे ड्रग सेफ है, मैंने ये स्टडी किया. जब आप उसको रिव्यू करेंगे तो मैक्सिमम दे ओनली इवैल्यूएट 10 to 15 पैरामीटर्स, नॉट मोर देन दैट. देन हाउ यू गेट द फुल प्रूफ कि ड्रग इज 100% सेफ. ओके, so that's why for safety concern you have to include as many as parameters. And previous to those, the WHO guideline also there and CCRS guideline, but CCRS guideline extracts the materials from this WHO and OECD guideline. CCRS ने खुद ने नहीं बनाया, उससे एक्सट्रैक्ट किया हुआ है, एडॉप्ट किया हुआ है. लेकिन अल्टीमेटली WHO का गाइड, अगेन WHO गाइडलाइन इज वेरी ओल्ड, 1993 का है, तो उसमें बहुत अपडेट की जरूरत, उसके बाद कोई उसका अमेंडमेंट नहीं है. तो मोस्टली लोग अभी आजकल शेड्यूल वाई, इस तरफ शेड्यूल वाई है और एक गाइडलाइन है, ये वाला जो दिखा रहा हूँ ये शेड्यूल वाई है. शेड्यूल वाई भी प्रिवेलेंट है इंडिया के अंदर फॉर सेफ्टी असेसमेंट, प्री क्लिनिकल एंड क्लिनिकल ट्रायल. तो OECD की जब बात करते हैं तो 420, 423, 425 ये 3 गाइडलाइंस है फॉर द इवैल्यूएशन ऑफ एक्यूट टॉक्सिसिटी. उसमें 5 एनिमल पर ग्रुप, 3 डोज लेवल है, 4 डोज लेवल है, 5, 50, 300 एंड 2000. मोस्टली ये रेड लाइन जो मैंने दिया है ना, ये वाला मोस्टली ट्रेडिशनल सिस्टम में मैं 20 साल से काम कर रहा हूँ, आज तक हमने जिसका भी इवैल्यूएशन किया ना सीधा 2000 पे ही करते हैं. क्यों 2000 पे? क्योंकि ड्रग है ना, अगर बाई चांस हाईली पोटेंशियल है, हाईली टॉक्सिक है, देन आपको लोअर में जाने कीजिए, नहीं तो डायरेक्टली आप यूनिट पे टेस्ट पे चेक कीजिए और सीधा रिजल्ट कीजिए. अभी जैसे मेरे पास एक स्टडी चल रहा था गुरुजी का, उसके गुरुजी पत्र ज्यादा प्रिस्क्राइब नहीं आएगी, गुरुजी स्टेम ही चलता है. तो गुरुजी पत्र का उन्होंने कंसेप्ट लिया था कि गुरुजी पत्र को हम ट्राइकोन्सिस बोलते हैं ना, क्या है, पटोल पत्र को रिप्लेस, मतलब उसका सब्स्टिट्यूशन की बात चल रही थी. ठीक है, तब गुरुजी पत्र और पटोल पत्र की जो दोनों की ऑलमोस्ट सिमिलर, तो उसका उन्होंने सेफ्टी ट्रायल्स किया. लेकिन अभी बात समझिए कि गुरुजी का ह्यूमन डोज 3 to 6 ग्राम, तो जब 6 ग्राम की बात करेंगे तो एनिमल डोज कितना आएगा, 540 मिलीग्राम. अभी उसका 5 टाइम्स तो पॉसिबल नहीं, क्योंकि 2000 इज इक्विवेलेंट टू 22 ग्राम इन ह्यूमन बीइंग ऑलमोस्ट. अभी 22 ग्राम पे अगर कोई ड्रग टॉक्सिक इफेक्ट नहीं है तो आप डायरेक्टली रिपोर्ट कर सकते हैं कि ड्रग इज सेफ, क्योंकि 25 ग्राम से ऊपर तो फिर नाश्ता हो जाएगा ना. इसलिए अल्टीमेटली 25 ग्राम तक ड्रग सेफ है तो आप सीधा रिपोर्ट kg is equivalent to 20 to 22 gram 0.5. 5000 तक तो बहुत एक्सेप्शनल केस में जाना है, बिल्कुल नहीं जाना। तो उस केस में आप सीधा लिमिट डोज पे काम करिए। 1000 mg पर केजी आपका हायर डोज पे है, यहाँ पे 1000 mg पर केजी है और एक्यूट में 2000 mg पर केजी। लोअर में जाने की जरूरत नहीं। कोई लोग बोलते हैं नहीं मैं 540 दूंगा। आजकल क्या हो रहा है, तो एक्यूट का भी ट्रेडिशनल का जितना पार्ट देखेंगे ना, किसी डीआई के पास अगर क्लेम आया होगा तो पता होगा वो लोग क्या करते हैं, मैंने 100 पे काम किया, 200 पे काम किया, 400 पे काम किया, 800। अरे भाई 100 का जरूरत क्या है, उसका और क्लिनिकल का कोई लेना देना ही नहीं। क्लिनिकल का डोज आपका 1000 है, तब ऑलरेडी उसका क्लिनिकल डोज इतना बड़ा है कि 100 पे काम करने की जरूरत है। और दूसरा अगेन और एक मजे की बात करिए, मैं इस तरफ बोला, अभी उल्टी साइड बोलता हूँ। मोस्ट ऑफ एफिकेसी है ना, लोग 400 मिलीग्राम पे करते हैं एक्सट्रैक्ट की, 100 200 400 मिलीग्राम। अभी बताइए 400 मिलीग्राम जब एक्सट्रैक्ट एनिमल में काम करेंगे तो ह्यूमन डोज उसका होगा भाई 4 से 5 ग्राम। तो वेदर एक्सट्रैक्ट 400 मिलीग्राम एनिमल ट्रायल करना जरूरी है? नहीं है, बिल्कुल। एक्सट्रैक्ट लेना है तो 50 से स्टार्ट करना पड़ेगा, 50 तो 200 वहां जाना पड़ेगा, क्योंकि 400 कभी भी आप कोरिलेट नहीं कर पाएंगे एनिमल ह्यूमन ट्रायल खास करके। तो इस पे में आपको डोज सिलेक्शन बहुत इंपॉर्टेंट क्राइटेरिया है फॉर द सेफ्टी ट्रायल। तो हाँ ओके नो, ये थोड़े से भस्म प्रिपरेशन, क्योंकि भस्म जो ऑलरेडी हमने कंप्लीट किए हुए हैं अपने इंस्टीट्यूट में, और शोधन प्रोसीजर, ये सारी प्रोसीजर है जो बहुत इंपॉर्टेंट है, खास करके ट्रेडिशनल सिस्टम ऑफ मेडिसिन में। यू नीड टू बी वेरी केयरफुल व्हाइल डिजाइन योर वर्क विद द सेफ्टी ट्रायल्स। Okay, these are the certain published literature from my side for the concept of, you see, the acute repeated dose toxicity of Rasparpati, then Tamrabhasma, then Amritikaran concept in the Tamrabhasma, and that study again we extrapolate for its hyperleptic anti-inflammatory activity and for clinical trials. And these the 5 drugs allotted to our institute, which are actually made by JAMA, and these are the guidelines for mutagenicity test. We conducted even 6 to 7. I started the work to 7 भस्म for जीनोटॉक्सिसिटी प्रोफाइल, क्योंकि कोई भी जीनोटॉक्सिसिटी प्रोफाइल भस्म का ही आजकल नहीं कर रहा है। हमने स्टार्ट किया और ऑलमोस्ट 6 भस्म कंप्लीटेड, रिसेंटली कंप्लीटेड आनंद भैरव रस and Rashmani KVC, and we completed Nag Bhasma as whole. You see, Nag Bhasma prepared by 7 food, 10 food, अलग अलग हमने मीडिया लिया, उसका पूरा केमिकल प्रोफाइल देखा, और भस्म भी उसका पूरा मोनोग्राफ पब्लिश हो गया है। ओरल टॉक्सिसिटी स्टडी, जीनोटॉक्सिसिटी, एंटी हाइपरग्लाइसेमिक, एंटी डायबिटिक, प्री क्लिनिकल एंड क्लिनिकल ट्रायल। तो in my first slide itself I told you, सिंगल कंसेप्ट कभी भी आयुर्वेद को हेल्प नहीं करेगा। कैरेक्टराइजेशन पार्ट से लेके क्लिनिकल पार्ट पूरा अगर करोगे तो ही you have the words to tell something. Okay, writing for the experimental research work मैंने जैसे डिस्कस किया, एसओपी से लेके पूरा आपको देना है, तो ही सेफ्टी ट्रायल अच्छी जर्नल में पब्लिश होती है। तो आप एसओपी, कैरेक्टराइजेशन पार्ट, इट्स प्रिपरेशन पार्ट, आईसी, देन कंट्रोल एंड व्हीकल कंट्रोल रखना बहुत जरूरी है। कोरिलेटेड एक्स्ट्रा कोरिलेट टू क्लिनिकल रिव्यू, एंड इसका मैं एग्जांपल सब जगह देता हूँ, तो 2 मिनट दूंगा। ये बहुत इंपॉर्टेंट पार्ट है कि देखो दोनों इक्वल दिखने वाले ड्रग है, एक है गुडुची, दूसरा है जस्टिसिया क्रिप्टोस्टेजिया ग्रांडिफ्लोरा। गुडुची सेम जैसा है, जामनगर में पूरे एरिया में ये दोनों साथ में चढ़े हुए। गुडुची जितना सेफ है उतना ही क्रिप्टोस्टेजिया टॉक्सिक है, इतना टॉक्सिक कि गुडुची, ये क्या हुआ कि एक दिन रात को फिजिशियन का मुझे फोन आ गया, सर आपका गुडुची कोई पेशेंट खा लिया, एक आदमी का डेथ हो गया, दूसरा आदमी आईसीयू। मैं बोला सर मैंने ही आपको कोविड के टाइम पे, तो फिजिशियन में जामनगर में सब लोगों को गुडुची की तबीयत हम देते थे। मैं बोला सर पॉसिबल नहीं है, वो मुझे ही फोन किया क्योंकि मैंने ड्रग दी। तो बोले सर कुछ तो है कि भाई एक आप तुरंत हेल्प कीजिए, इसमें क्या कर सकते हैं। तो मैं बोला सर वो भेजिए, हम करेंगे। दूसरे दिन वो रिलेटिव सारा मटेरियल लेके आ गए, तो सब बच्चों को दौड़ाया कि भाई है क्या। सब बच्चे आसपास गए, द्रवगुण वाले ढूंढ के लेके आए इमीडिएटली। पास में जो एरिया है पूरा दोनों साथ में चढ़े हुए। अभी उस दिन वो बंदे ने क्या किया, गुडुची की जगह इसका काट लिया दातुन करने के लिए। उसने तो लिया, फ्रेंड को भी खिलाया, तो जिसने लिया था वो बच गया, लेकिन जिसने खाया, दूसरे को जिसने दिया, वो राजकोट में उसको डेथ हो गई, क्योंकि इमीडिएटली हाइपरकैलेमिया होता है, और सिर्फ इतनी कारण से हाइपरकैलेमिया होता है ना। तो वो जो बच गया ना, वो भी कम से कम 15 से 30 दिन तक आईसीयू में रहा, और बीच बीच में सोप देना पड़ा, तब जाके बाहर आया। तो दिस इज बेस्ट एग्जांपल कि अगर आप रॉन्गली ड्रग को आइडेंटिफाई करते हैं, इसलिए इसका एग्जांपल मैं हमेशा इसलिए देता हूँ कि कई बार गुरु जी के ऊपर आर्टिकल पब्लिश होते रहते हैं ना, और मेरे से पहले उन्होंने भी रख दिया था अपने ग्रुप में कि भाई गुरु जी खाने के बाद ये, क्योंकि उसके भी ग्रुप्स है ना। फिर इमीडिएटली मैंने बोला कि भाई ये पॉसिबल नहीं, फिर उन्होंने रिवाइव भी किया कि एक्चुअली इट वाज नॉट गुरु जी, इट वाज the triptostage Jagdani product. This is the example of wrongly choose the drug. And you see the recently WHO center at Jamnagar for the its policy, and already the center started, interim center started, and naya center बनने जा रहा है. And our institute already collaborating Center for Traditional Medicine in case of certain TOR for safety monitoring, for the building research capacity, and the WHO tool strategy, and many more. So I here complete my lecture. So thank you.
Thank you, Dr. Mukesh.
सर आपका लेक्चर जो है टॉक्सिकोलॉजी और फार्माकोलॉजी में बहुत मतलब ब्रिलियंट और एक्सीलेंट लेक्चर। जब हम अपना पीजी कर रहे थे फार्मास्यूटिकल से, तो उस समय ये फार्माकोकाइनेटिक और डायनेमिक्स पढ़ रहे थे। आज आपने फिर दोबारा से रिवाइव करा दिया हम लोगों को, जो भी है तो बहुत अच्छा है। और एक्चुअल में क्या है, जो मॉडर्न फार्माकोलॉजिस्ट होते हैं वो हमारे ट्रेडिशनल सिस्टम को समझ नहीं पाते हैं। कैसे भस्मा का जैसे आपने बताया ना कि जब तक आप प्रॉपर सॉल्युबिलिटी होगा ही नहीं तो प्रोडक्ट में उसका इफेक्ट क्या है, तो वो एक आज हमारे उसमें सीखने की ज़रूरत है। आप लोग यहाँ से जा रहे हो, जो लोग काम कर रहे हो प्रिपरेशन पे या ड्रग बना रहे हैं या उसके आगे स्टैंडर्डाइजेशन कर रहे हैं, तो इन बातों का जरूर ध्यान रखें कि उसकी सॉल्युबिलिटी हो रही है या मैच इंटरसेप्ट हो रहा है कि नहीं हो रहा, बॉडी के साथ इंटरेक्शन क्या है, ये स्टडी भी डालेंगे तो ज़्यादा अच्छा रहेगा। जो रिजल्ट आएंगे फिर प्रॉपर रिजल्ट आपको मिलेंगे एक्टिविटी के बेस। थैंक यू। अभी कोई क्वेश्चन हो डॉक्टर साहब से रिगार्डिंग फार्माकोलॉजी का, तो लेक्चर से संबंधित, तो आप लोग पूछ सकते हैं।
Sir, this is regarding the heavy metal use of Rasaushid in OPD. So what is your stand, sir? How we take it out? Because so far I have not, since almost 25 years I'm into clinical research and I'm an Ayurvedic practitioner. I have not seen any drug inducing the nephrotoxic or the toxicity and safety profile are very normal.
एक्चुअली हेवी मेटल्स जब फ्री फॉर्म में है तब की बात है। ये भस्मा है वो एक्चुअली हेवी मेटल्स नहीं है। भस्मा हेवी मेटल्स नहीं, भस्मा तो एक प्रिपरेशन, आयुर्वेदिक फॉर्मुलेशन। हेवी मेटल्स तो कब बात करेंगे, तो जब वो बिल्कुल फ्री फॉर्म में, फ्री फॉर्म एज पर जो हम बोलते हैं ना ऑर्गेनिक इनऑर्गेनिक मटेरियल जैसे वो है, वो टॉक्सिक।
But if it is, that is the case, sir, then they ask us to use Ribonacircuit only for this many days. Like that, if you have some,
ये इसलिए बताया है कि वो हाईली पोटेंशियल ड्रग है। मैं आपको बोलूँ कि जैसे मेरा ही मैं अगर एक्सपीरियंस बोलता हूँ कि जितने भी हम कई बार 90 डेज स्टडी में जाते हैं, 120 डेज स्टडी में जाते हैं, तो इसलिए बोला गया कि शेड्यूल E1 ड्रग, the fish drug, all metal mineral preparation drugs, those drugs are very potential drugs. और हमेशा एक चीज ध्यान रखें, जब ड्रग एफिकेसी हाईली पोटेंशियल है, बहुत हाईली पोटेंशियल, साइड इफेक्ट या उसका जो सेफ्टी कंसर्न भी जनरेट होना ही होना है। रिबोन कीर्ति रस है, एक दिन में आपको तुरंत सुखा देगा पूरा सिस्टम, तो स्वाभाविक है कि कहीं ना कहीं वो एक जगह पे उसका सेफ्टी सॉरी एफिकेसी आ रहा है, तो कहीं वो दूसरे जगह, क्योंकि ये देखो बायोलॉजिकल सिस्टम में सारे रिसेप्टर्स है, दुनिया भर के प्रोटीन है, दुनिया भर के टारगेट, एक टारगेट पे पॉजिटिव आ रहा है, तो कहीं ना कहीं सेम टारगेट लोकेटेड दूसरी जगह पे भी है। फॉर एग्जाम्पल, उसके लिए मैं मॉडर्न का एग्जाम्पल दूंगा, तुरंत आपको समझ में आएगा। देखिए जितने भी कफ एंड कोल्ड वाले ड्रग्स है, ओके, आप लॉन्ग टर्म यूज करेंगे ना, टेकिकार्डिया डेवलप हो जाता है। क्यों डेवलप होता है? क्योंकि बीटा 2 जो आपका रेस्पिरेटरी में वो रिलैक्स होता है, लेकिन अल्टीमेटली बीटा 1 जो आपका कार्डियक पे वो स्टिमुलेट होता है। तो कई बार ऐसे मिसअंडरस्टैंड डेवलप हो जाते हैं, जैसे 1 महीना खाया तो शॉर्ट टर्म टेकिकार्डिया हो गया। सेम वे अगर हार्ट वाला ड्रग लेते हैं तो आपका रेस्पिरेटरी रिलैक्स होता है। तो जरूरी नहीं ड्रग तो
no, sir, this is only symptomatic based. What is other the biochemical changes or any safety profile?
बायोकेमिकल चेंजेस यू हैव टू डेवलप दोस चेंजेस, क्योंकि ये बहुत जरूरी है। उसमें इसीलिए मैं बोल रहा हूँ जब सेफ्टी कंसर्न की बात करते हैं ना,
क्योंकि आयुर्वेद में हम थोड़ा अदरक दे दिया तो फिर उस लोग को हिट हो जाता है, ऐसे भी है, वो तो सिम्टोमेटिकल है। व्हाट इज द इफेक्ट ऑन सेफ्टी प्रोफाइल, लाइक
प्रोफाइल इसीलिए मैं बोल रहा हूँ कि सेफ्टी प्रोफाइल डॉक्यूमेंटेड है ही नहीं, करना चाहिए। इसलिए हम देखो, मैं बताया बहुत सारे फॉर्मुलेशन का मैंने नाम बोला, उसको आपको सिस्टमेटिकली इवेल्यूएट करना पड़ेगा, नॉट ओनली प्री क्लिनिक, अल्टीमेट एम इज क्लिनिकल। तो क्लिनिकल में भी जितने भी क्लिनिकल ट्रायल्स करते हैं, वो क्लिनिकल ट्रायल्स में क्लिनिकल ट्रायल्स के साथ सेफ्टी मॉनिटरिंग के भी डेटा उनको साथ में रखना चाहिए। फेज 4 ट्रायल बहुत जरूरी है, और वो मिस कर रहे हैं।
Usually we keep LFT, RFT, and we keep
बहुत जरूरी फेज 4 ट्रायल है ना सर, डेवलपिंग कंट्री एंड अनडेवलप्ड कंट्री में फेज 4 ट्रायल को मिस कर रहे हैं। डेवलप्ड कंट्री में फेज 4 क्लिनिकल ट्रायल का कंसेप्ट हम करते ही नहीं, ना डॉक्यूमेंट करते। और फेज 4 क्लिनिकल ट्रायल बहुत इंपॉर्टेंट है। फेज 4 क्लिनिकल ट्रायल में क्या, पोस्ट मार्केट सर्विस, और उसमें सबसे बड़ा रोल किसका है, फिजिशियन। यहाँ पे फेज 4 क्लिनिकल ट्रायल हम क्यों नहीं देख पाते? पेशेंट को इफेक्ट नहीं आया तो पेशेंट ही डॉक्टर को चेंज कर देता है, दवाई चेंज कर देता है, तो आपको एग्जैक्टली समझ में ही नहीं आएगा कि उसके जो टॉक्सिसिटी आ रहे हैं, वो दवाई आगे वाला दिया था उनसे आ रहा है, या इश्यू आ रहा है, वो क्या आ रहा है। और दूसरा, डॉक्टर के पास भी उतना टाइम नहीं है कि हर एक चीज को नोट डाउन करे। अगर ये कंटिन्यूसली प्रैक्टिस में आ जाएगा ना, तो सर मतलब 100% डॉक्यूमेंटेशन बहुत जरूरी है। डॉक्यूमेंटेशन के बिना काम नहीं
सर आपका एक्सपीरियंस हिसाब से वो रसौषधि कैसे एक्सक्रीट हो सकता है?
सर, ये एसिमुलेट होता है, अब तक हमने ये देखा। वो मोस्टली मेटाबोलाइज बहुत कम मेटाबोलाइज होता है, मोस्टली एसिमुलेट होता है। अगर वो बहुत छोटे पार्टिकल में एसिमुलेट, मतलब वो कहीं ना कहीं जाके वापस एक्सक्रीट होकर निकल जाता है। और मैंने देखा है, जितने भी हमने भस्म प्रिपरेशन पे काम किया है, आफ्टर रिकवरी ग्रुप हम सब में रखते हैं। रिकवरी ग्रुप सब में, रिकवरी मतलब 90 डेज तक रख देना है, फिर 30 डेज ऐसे ही छोड़ देना, कुछ नहीं देना। ओके, और 30 डेज के बाद हम उसको सेक्रीफाइस करके अगर हम हिस्टोपेप करते हैं ना, कहीं पे भी डिपोजिशन नहीं मिलता। लेकिन वही 90 डेज में मैंने देखा है, कई बार क्या होता है, हमने तुरंत किया ना, तो कई बार छोटा मोटा डिपोजिशन दिखता है, लेकिन वो जैसे ही आएगा वो स्ट्रेट हो जाए। तो इसलिए एसिमुलेशन होता है मोस्टली, एसिमुलेशन पार्ट बहुत। पर एसिमुलेशन कब होगा? अगर वो नैनो पार्टिकल है तो ही होगा, नहीं तो नहीं होगा। अगर माइक्रो या उससे कम में है तो कहीं ना जाके फंस जाएगा, और फंस गया तब वो टॉक्सिक हो जाए। तो इसलिए ये चीज बहुत इंपॉर्टेंट है। और क्लिनिकल सेफ्टी तो है ना सर, फिजिशियन अगर थोड़ा अवेयरनेस और ये सब डॉक्यूमेंट करेंगे ना, तो ही ये पॉसिबल है। क्लिनिकल ट्रायल से ही आपको पता चलेगा, और इसके लिए सबको ध्यान रख के डॉक्यूमेंट करना बहुत जरूरी है।
Sir, I would just like to add to his point. अगर आप accumulation देख रहे हो in animal studies, right? Improperly prepared rasa preparation, you can see accumulation at the level of kidney. So if you're going to sacrifice the animal's money to do a kidney slide study, आप देखोगे तो mercury and everything you can get. So more than it won't accumulate in liver or brain, but majorly first accumulation will be in the kidney. सर मेरा एक क्वेश्चन है, जो आजकल जो प्रोप्राइटरी मेडिसिन आते हैं, फॉर एग्जांपल आई एम जस्ट सेइंग टॉप ऑफ माय हेड, महाराष्ट्र कषायम के साथ एकांगवीर रसन समीर पन्नागर ऐसे मिक्स करके देते हैं। आई मीन 3 ऑफ देम आर इंडिविजुअल क्लासिकल ट्रेडिशनल मेडिसिन, तो उसके मुताबिक तो आपको एफिशिएंसी नहीं लेना पड़ता है, पर जब ये ऐसे कॉम्बिनेशन में लाते हैं तो हाउ डज द रूल अप्लाई?
कॉम्बिनेशन का हमारा कोई भी स्टडी नहीं है एक्चुअली, इसीलिए मैं बताना चाहूँ, इसलिए हाईलाइट ज्यादा कर रहा हूँ कि द्रव्यगुण जो ब्रांच है, आयुर्वेद आप शायद जानते हैं, द्रव्यगुण ब्रांच में बहुत अच्छा कंसेप्ट है रत्न। जब तक कॉम्बिनेशन में प्रॉपरली, अगर या तो उसको स्टडी करना पड़ेगा या प्रॉपरली, क्योंकि इंडस्ट्री में कहीं पे भी अभी नो डाउट, अभी तो देखिए एमडी वाले या बीफार्म एमफार्म वाले जाने लगे। पहले तो क्या था, आयुर्वेद ड्रग इंडस्ट्री को कोई भी ड्रग बनाना है कोई बड़ी बात नहीं थी, सिंपल एक 50 रुपए का फॉर्म है, उसको फिलअप करो, फॉर्मुलेशन बनाओ, कोई अप्रूवल नहीं, साथ में कुछ देखा नहीं, सिंपली गेट द लाइसेंस, है ना। और लोग बनाते थे, कौन बताएगा? जब आप पेशेंट को बता देंगे, पेशेंट बताएगा रिजल्ट आया या नहीं आया। तो ये मेजर लेक्यूना, और नो डाउट, लेकिन आजकल जो है ना, बहुत इसमें चेंजेस आए हैं लास्ट 5 से 10 साल, खास करके कोविड के बाद बहुत सारे चेंजेस आए। नाउ रेगुलेटरी एजेंसी आल्सो टेक द नेसेसरी एक्शन कि इतना बोटेनिकल ये होना चाहिए। तो एक खास करके जो आपका पॉइंट है ना, इंफेक्शन वाला पॉइंट, उसमें आज तक कोई इतना काम नहीं हुआ, उसको भी एक साइंटिफिक काम उस पर बहुत अच्छे से डिजाइन करना चाहिए हर एक ड्रग का, कि किसको किसके साथ देना चाहिए। कहीं साथ में दे रहे तो एफिकेसी ज्यादा नहीं बढ़ रहा है, कम नहीं, क्योंकि देखिए इसका बेस्ट एग्जांपल कुरकुमिन। कुरकुमिन वेल स्टडीड, बिकॉज आई शहर के बहुत इंस्टीट्यूट ने कुरकुमिन को स्टडी कर लिया कि भाई कुरकुमिन जब एंटीटीवी ड्रग के साथ देते हैं तो जो रिफार्मेसिन आइसोनिजम का बायोबिलिटी बढ़ जाता है ना। तो सेम वे बाकी ड्रग्स का भी लीवर के एंजाइम्स को इनहिबिट कर रहा है या लीवर के एंजाइम्स को एनहांस कर रहा है। कोई भी ड्रग लीवर के एंजाइम्स को इनहिबिट करता है या उसको स्टिमुलेट कर रहा है, इसका मतलब वो ड्रग का पक्का रोल है मेटाबॉलिज्म को या तो बढ़ाएगा या घटाएगा। तो जो साथ में ड्रग दे रहे हैं उसका या तो मेटाबॉलिज्म बढ़ेगा या घटेगा, तब ये देखना बहुत जरूरी है। तो इसलिए कोई साइंटिफिक ऐसा रिपोर्टेड मटेरियल नहीं इसको किसके साथ, बट हाँ क्लासिक्स में फॉर्मुलेशन प्रैक्टिस में लोग करते हैं, लेकिन उसको आप जो एक्चुअल प्रैक्टिशनर है वही इसमें ज्यादा रोल प्ले कर सकते हैं कि भाई कॉम्बिनेशन जैसे मॉडर्न का तो ऑलरेडी है जिसको इसके साथ, और आए दिन आप देखेंगे कि जो अपना रेगुलेटरी है सीडीसी तो वो आए दिन फॉर्मुलेशन को बैंड, अभी रिसेंटली 50 फॉर्मुलेशन को बैंड कर दिया रिसेंटली, भाई इसको विड्रॉन कर लो, एक साथ नहीं दे सकते। मोस्ट ऑफ कफ एंड कोल्ड का मैं देख रहा था, आप देखो कोई भी फॉर्मुलेशन कैफीन होगा, पैरासीटामोल होगा, फिर हाँ 4-5 साथ में ही रहता है। तो इसीलिए मोस्टली वो तो 3 से ज्यादा अलाउड नहीं करते, 10 तो बहुत दूर की बात है, 2 या 3 से ज्यादा अलाउड नहीं करते। तो इसलिए थोड़ा स्टडी इसके ऊपर इनिशिएट होना चाहिए और इसमें ड्रग इंटरेक्शन पार्ट को बहुत डिटेल से स्टडी करना
How can we consider ADNE for homeopathic dilution?
है। डाइल्यूशन उसके लिए बायोलॉजिकल मार्कर्स होगा ना मैडम, जानना चाहिए।
But there is so much in dilution form, so biomarkers तो फिर उसमें detect ही नहीं होगा।
पहले केमिकल मार्कर नहीं आ जाएगा, इवन माइक्रो नैनो में भी आ जाता है, अगर आपके पास अच्छा ये है, वो आपकी बात बिल्कुल सही है। मैं highlight इसलिए करूंगा, क्योंकि देखो कई बार हम क्या करते हैं बाहर से ड्रग को ओरल अवेलेबल ओरली देते हैं, अभी वो बॉडी में अभी ब्लड है 5 to 6 वहां जाके वो घूमे, ब्लड से आप निकालते हैं 1 ml 2 ml, तो जो दिया और ब्लड में 1 या 2 ml में लिया तो कितना डाइल्यूशन हो जाएगा। तो उसके लिए आपको हाईली सोफिस्टिकेटेड टेक्निक, फिर आप क्या करना है उसके लिए मेथड नहीं है, इसके ऐसे डेवलपमेंट का बहुत इंपॉर्टेंट पार्ट है वो ड्रग डिस्कवरी प्रोग्राम ऐसे डेवलप। और इसके लिए मैं एग्जांपल एक हमने प्लान किया था ट्रिपल अपाइजिंग ऑफ मस्ती एंड और दूसरा प्लान किया था ब्राह्मी ग्रिप टू नशे एंड और ब्राह्मी ग्रिप जब नशे से दिया और ओरली दिया तो दो हमने बायोलॉजिकल मार्कर्स ग्लूकोपासाइड A3 और एक्कोपानाइन ऐसा कुछ दो मार्कर्स फाइल ब्रेन के अंदर कॉन्सेंट्रेशन देखा और सीरम में देखा। अभी मेजर इश्यू क्या आता है कि भाई आपने दे दिया, अभी सीरम से उसको हम कैसे निकालेंगे, क्योंकि अल्टीमेटली तो आपको कलरिंग मेथड या कोई मेथड में जाना है, तो वो मेथड तो हमारे पास है नहीं, तो ट्रायल एंड एरर करके वो मेथड को चेक किए और उसका इवोल्यूशन मेथड। और एक एग्जांपल दूंगा, तुलसी का हमने बिंदु बनाया, तो स्टूडेंट बनाया, उसको राइनिटिस के लिए था क्लिनिकल ट्रायल, फिर और नशे से किए थे एक ओरली, तो सेम वे उसको देखना था। अभी हम क्या करेंगे सीरम डाल के सीधा फॉर एग्जांपल करने जा रहे हैं, तो जिसकी वजह से उसको मोलेक्युलर कंस्टिट्यूएंट है आपके तुलसी में तो सारे उड़ रहे, पॉसिबल नहीं है। जब डायरेक्टली कोलम चौक हो जाएगी, फिर करे तो क्या करे, मतलब सौराष्ट्र में एक अच्छा सेंटर है मेरा ब्रदर उधर है, फिर उनके साथ बहुत सारा डेलिब्रेशन करने के बाद लास्ट में हमने एक इंटरमीडिएट मेथड निकाला कि भाई ये इंग्रेडिएंट ऐसी मेथड से जाएंगे तो डिटेक्ट हो जाएगा। लास्ट में 10 ऐसे टारगेट्स को हमने फिक्स किया बायोमार्कर कि भाई ये मेथड तो ये 10 पक्का डिटेक्ट हो जाएगा, फिर वो 10 को हमने डिटेक्ट। तो ऐसा आपको कुछ ना कुछ मेथड या समथिंग आपको करना पड़ेगा बाय बाय द हेल्प ऑफ केमिस्ट्री, क्योंकि यहाँ पे मेजर रोल केमिस्ट्री का है, क्योंकि वो बायोलॉजिकल मार्कर इसलिए मैं बोला केमिकल मार्कर ऑलवेज नॉट बायोलॉजिकल, क्योंकि केमिकल बायोलॉजिकल सिस्टम क्या करेगा हमें पता है, ठीक है, तो आप कर लीजिए उसको जाता है बहुत बढ़िया से है। हमने अभी रिसेंटली नाइपर का प्रोग्राम था खास
Now,
May
sir will give you his email ID and phone number also, so you can interact with him personally because we are late for the valedictory program. So I will request SC Burma sir to kindly felicitate our guest with the memento of HPL and with the certificate.
वर्मा जी मेरे बहुत अच्छे फ्रेंड है, हमने सीसीआरएस में कैरियर एक साथ स्टार्ट किया था 2006 तक का बहुत सहयोग रहा केमिस्ट्री में भी, क्योंकि हम दोनों सीसीआरएस में काम करते थे तो केमिस्ट्री पॉइंट सब देख के हमें थोड़ा गाइड भी करते थे।
कम्बाइंड थेरेपी है, फार्माकोग्नोसी का एक पर्सन होना चाहिए, केमिस्ट्री है, एक फार्माकोलॉजी, देन यू कैन डेवलप ए डेफिनेटली ए न्यू नॉवल फॉर एनी स्टडी, ठीक है। और ओवरऑल क्लिनिकल एफिकेसी के लिए आपको एक फिजिशियन की जरूरत पड़ती है, तो 4 का कॉम्बिनेशन अगर बना लें तो बहुत अच्छा रिसर्च हो सकता है हर यूनिट में। Now just we want to move just in education for packet 2 auditorium hall where we met previously in inaugural function.